All right. So, part three, the heart. This is the final part to this cardiovascular lecture. And this is probably the most important part. This is the organ that does the pumping, right? So, here are your learning objectives, right? We're going to start by looking at the anatomy of the heart, the overall anatomy. So, where is the heart in the body? Here is a real heart, a picture of a real heart. Here is where you would locate it, okay? So, this is actually called the apex of the heart at the bottom. This is the apex. This points inferiorly, meaning downwards, towards the left hip, right? It's tilted a bit to the left. And this part here is what contacts the chest wall, right? It's the point of maximum intensity in terms of the contraction. This is where you're really going to squeeze, and you squeeze the heart up like a tube of toothpaste. And this is why you could feel your heart beat against your chest. If you put your hand over the left part of your chest, you will feel the apex beating against the wall. It contacts the chest wall. Why is it called the apex? Usually an apex is like at the top, right? Actually, an apex just means the pointy part of a triangle, like the highest point of the triangle. And on a mountain, the highest part of the triangle, the pointy part is at the top, right? But the heart actually is inverted, right? The pointy part of the mountain is actually at the bottom, right? This is the apex. The base of the heart is the more flat and broad aspect of the heart. This is all considered the base. Think of a triangle, the base, the apex at the top, right? So you could almost think of it like this triangle over here, right? With the base being here and the apex being at the tippy top over here. And that's the base is the part that's directed towards your right shoulder, okay? And the point of the base is to have all the vessels that are exiting and entering at that point, okay? So that's the big positioning of the heart that we see. I want to talk about what the external coverings of the heart are. So first we have the fibrous pericardium, okay? So this is a loosely fitting superficial part of layer that's composed of this very dense connective tissue. So it's this outer part here of connective tissue, and its function is to protect the heart, to anchor the heart to surrounding structures, right? This heart is not just flopping around, it's connected to actually the mediastinum. It's this connective tissue all around. So this connects to this other connective tissue. And also, this fibrous pericardium prevents the overfilling of the heart with blood. Because if the heart could fill as much blood as possible, right, it could expand and burst. Just like a balloon. You know how a balloon, you could blow into the balloon until it bursts? Well, thanks to the fibrous pericardium, that doesn't happen with the heart. Because it's encapsulated. Imagine blowing a balloon in a cement in this cement block, right? It would stop eventually, and you wouldn't be able to put more air in it than you need to, right? Because it would stop and it would push against the cement. Same thing for the heart here. The heart can't overfill with blood and expand too much because of this fibrous pericardium, which is tough. Then over here you have this serous pericardium, okay? So you have an inner layer and an outer layer, right? This is a thin, slippery, two-layered serous membrane, right? There's the parietal layer and the visceral layer. So the parietal layer lines the internal surface of the fibrous pericardium. So it's this is the internal surface of the fibrous pericardium and attaches to very large arteries that are exiting the heart at the superior margin. So if you follow it through this parietal layer, you will see that there's these attachment points to these vessels over here. The visceral layer forms the epicardium. Another layer for the visceral layer, the one that's touching it, is called the epicardium, right? So it forms the part of the heart wall, the epicardium. And remember this serous pericardium, right? The parietal layer is the outer layer. The visceral means it's touching the viscera, the actual organ, right? So, and there's a space in between, right? I'm going to talk about that pericardial cavity that's in between. The pericardial cavity is between the parietal and the visceral layers of the serous pericardium. There's this space over here, right, in between. And this is filled with what we call pericardial fluid. So if the pericardial fluid, it's what fills the pericardial cavity. It's a type of serous fluid, right? And it acts to lubricate the serous membranes. So it lubricates these two membranes on each side, and it allows the heart to glide freely, right? So it kind of makes a cushion, right? So that the heart can move and it won't jab into another organ on the side and damage itself, right? So it allows this, it's kind of like a protection, like a waterbed, right? This is a waterbed that's kind of protecting it. It's about 15 to 20 mils in volume, actually, of serous fluid inside of here. So I actually much prefer this picture. This picture is a bit hard to see the stuff that I need to. I really like this picture. Here's your fibrous pericardium, right? That tough external fibrous layer. Then you have the pericardial, the parietal pericardium over here, followed by the connected visceral pericardium here, touching the heart. And in the middle over here, you have the pericardial cavity. This is where the serous fluid is. I want you to imagine it filled with water. I want you to imagine it as an aquarium because it is closed. Here it looks two-dimensional, but it's actually three-dimensional. It's a little layer, a little aquarium, right? Fishies can swim in here. It's all connected and it's filled with this liquid that just stays there, right? It's like a spherical waterbed. And then over here, right, is our visceral pericardium. And then we have the heart wall, okay? The heart wall is the actual heart itself. These are just layers that are protecting from the outside, right? But these ones in darker red are actually the heart wall. And actually the visceral pericardium is part of the heart wall. But we call it something different when we refer to it as the heart wall. When we refer to it as the heart wall, we call it the epicardium, okay? So the heart wall has three layers: the epicardium, the myocardium, and the endocardium. Remember, the epi means the topmost layer, right? The externalmost, the top on top, epi. So this is the epicardium, the topmost. Then you have the myocardium. That's the thickest of the layers. This is full of myo, muscle, and it's also M for middle, right? So myo for muscle and M for middle. It's the middle layer of the heart wall. It's the thickest layer, full of muscle. This is what does this contracting, right? This is what squeezes and gets smaller. Think of the water balloon when you take your hands and you squeeze the water balloon. That's the myocardium that's doing that, the squeezing. And lastly, on the inside, touching this is the heart chamber where the blood is found. You're going to find some blood in here. The one touching the blood is called the endocardium. It's the innermost layer of the heart wall. So this is on the inside. Endo means inside layer. Epi means outermost, the top layer. So again, let me just go through what this pericardial cavity is to remind ourselves. In terms of the structure, right, this is a double layered pericardium. It has a visceral pericardium that is over here touching the heart. And there's this parietal pericardium on the outside. And it forms a space called the pericardial cavity. All of this here is like the pericardial cavity, which is filled with serous fluid. Okay, this is all filled with water. I want you to imagine in 3D, you taking your fist and punching into a balloon. This is all space, right? Closed in space. Imagine it filled with water now. right? And that's what the idea is of the pericardial cavity. The fluid-filled space, the pericardial cavity, has a few functions. The first one is it allows for some capacity for stretching, but it prevents overfilling of the chambers, right? So the blood can stretch a bit thanks to the waterbed, but it also stops it from overfilling too much. And secondly, it reduces friction between the heart and the surrounding organs. Because the heart is pumping, right, it might push a little bit into some areas, right? But thank goodness the heart isn't pushing and going into the connective tissue and injuring itself. There's this water bed to kind of protect it and reduce the friction. This is very, think of how much you can move on a water bed, right? It's very like, like you can move around a lot because of that watery substance, right? So actually it really helps to eases the heart's contraction so that it could do so in this space, this waterbed, right? So, as I said before, the heart wall is composed of three layers. We have the outermost layer, the epicardium. It's the outer layer of the heart wall, but the inner visceral layer of the pericardium, right? You could call it the visceral pericardium or the epicardium. It's the same thing. This middle layer here, I said it's the thickest layer made of cardiac muscle, right? And that cardiac muscle is there to contract and to pump the heart, right? It also contains a lot of blood vessels and nerves because you know that muscle constantly requires glucose, constantly requires oxygen, constantly requires nerves to go and talk to it. So this is our muscular layer, right? But it's cardiac muscle. And then we have an inner layer, the endocardium, on the inside over here. This lines the inside of the heart chambers, and it's very smooth. It's this small little layer, smooth, that reduces friction of blood for better pumping efficiency. Imagine this layer was jaggedy and all crazy connective tissue. The blood would not would kind of be turbulent inside of it. So it's nice to have this very smooth inner layer of the heart. Okay. As for the heart chambers, right? The vessels and circuits, right? We're gonna go through those now. I want to remind us of something very important we just saw recently. How do we name blood vessels in the heart? The arteries, right? Remember, arteries go away from the heart. A is away. So let's say the heart is here, it's sending blood away from the heart this way. The direction matters. Capillaries, they're the smaller vessels, right? This is where oxygen, nutrients, they exit the blood and come back in the blood. The waste and carbon dioxide comes back into the blood. And then that blood goes through the venules and goes to the veins to return. I really want to emphasize arteries versus veins. A is away from the heart. Veins pump blood towards the heart. Revient, venir au cœur. This is going back to the heart. This is the caution. I know in this picture, this is red, right? But arteries do not always carry oxygenated blood. They're not always red. Sometimes they're blue when they go to the lungs. Veins do not always carry deoxygenated blood. I know in this picture it's blue and it's deoxygenated. They just gave their oxygen to the tissues and brought back CO2. But we're going to see that that's not always deoxygenated, okay? So what are the three circuits of the cardiovascular system, okay? I want you to know these three circuits, right? Just a bit of fun facts before. The heart beats 100,000 times per day, pumping 8,000 liters of blood through its three circuits, right? That's a lot of blood. And what are these three circuits? These three circuits are the pulmonary circuit, the systemic circuit, which we talked a lot about already, and the coronary circuit. What is a circuit? The heart is the beginning and the end of each circuit. The definition of a circuit, right? In Montreal, we have Le Circuit Gilles Villeneuve. A circuit is a loop that comes back to its start point, right? It's where a circuit, by definition, means that where you start from, you go around and you come back to it, right? And where do you come back to in these circuits? It's the heart, right? The pulmonary circuit, poumon in French, means lungs, right? This is the lung circuit that carries blood from the heart chambers to the lungs. Okay, so here, do you see this circuit here? You have the blood going from the heart to the lungs over here and back to the heart chambers, back over here, right? So that's the pulmonary circuit going to the poumons, the lungs. The systemic circuit is actually multiple mini circuits, right? That carries blood from the heart chambers all the way to the body, the peripheral tissues, to the system. It's called systemic circuit. It carries it to all of the systems, down to your toes, up to your hair over here, right? So some of these circuits, some of these capillaries are down at your toes. Some of these capillaries are up at your head, okay? So they carry from the heart to the peripheral tissues and back to the heart chambers, right? If we go back over here, I'll show you a bit. In one of the, I did like to look at in the capillaries over here, right? You see these capillaries here, right? This is going to be the pulmonary circuit over here. You see that the blood vessels go to the lungs and then back over here to the capillaries, back to the heart, right? That's the pulmonary circuit. Whereas everything else here is actually pretty much the systemic circuit. So you could look at a systemic circuit going from the heart over here to these capillaries and then coming back through the veins and going back to the heart. Or you could think of this as going up here to the head, coming back through the veins and going back to the heart, right? That's also the systemic circuit. So there's actually many little mini capillaries in the systemic circuit, but we like to think of it as one. It's a lot easier. Okay, so let's continue where we left off. We have the pulmonary circuit, the systemic circuit, and lastly, we have a mini systemic circuit, but we call it a special name because it goes directly to the heart. That's called the coronary circuit. Anything coronary sounds like the heart, right? The coronary circuit, this is a bit weird. It carries blood from the heart, inside the heart, out from the heart chambers, out to the tissues outside of the heart itself. Do you see these little vessels here, red and blue? The heart is a muscle. It needs to feed itself, right? We feed the toes, we feed the hair, we feed the arms, the legs. We also need to feed the heart itself. It's really strange, but you will send vessels out, right? Let's say you're a cook at a restaurant, right? You feed everybody at the resto. Eventually you need to eat. So you will make food for yourself, right? Same thing here. The blood pumps everywhere to the body to feed those tissues, but it also needs to feed itself. So it sends a little artery out of the heart chambers to the tissues themselves. It feeds those cells and then it goes back through these veins and returns back to the heart. So there's a mini circuit that goes to the heart itself to feed the outer muscle of the heart. This is a very important circuit actually because the heart needs oxygen too. If the cardiac muscle doesn't have oxygen, it will die. If the cardiac muscle dies, it will not pump. If you don't pump, you will not be sending blood anywhere. It's like the chef, right? The cook. If the cook is not there, you are not sending food to yourself nor to anybody else in the restaurant, right? So that's the idea of these circuits here. So how does gas exchange occur at the capillaries, right? Well, here's my heart, right? In the pulmonary capillaries, we should have drawn some arrows. Actually, I'm going to do that with you now. The pulmonary capillaries, at this area over here, we will send to the lungs deoxygenated blood. It's blue. It's full of CO2, not full of oxygen, okay? And the point of going to these pulmonary capillaries is so that CO2 diffuses from the blood, from these vessels, out into the lungs, and then you will breathe out. Exhale that CO2. And then you'll go, and you will bring in oxygen from the lungs into the blood, okay? So you bring this deoxygenated blood to the capillaries, you put it out into the lungs to be exhaled, and you bring an inhale in oxygen so that that will enter into the capillaries. And look, now at this end, my blood is red, right? And so you will return to the heart, right? With this oxygenated blood ready to go. So the transport of respiratory gases, so first we pick up and remove these gases here, and then we transport these respiratory gases through the cardiovascular system, right? Using all these, this blood as a transport system through the highway, through these vessels over here, right? So over here you'll see we'll go back to the heart, right? And then we'll be pumped out this way through this tube. And we will go get transported to the systemic capillaries, okay? Let's say these are the tissue cells in your toes, right? So at this point, this oxygenated blood will go and diffuse from the blood into the tissues, right? The tissues will take and need oxygen, and the CO2 will diffuse from the tissue cells out into the blood. And that CO2 will be transported back to the blood. And that CO2 deoxygenated blood will go back to the lungs so that we can remove the CO2 and exhale it and take in new oxygen. Because these cells here, right, they're continuously metabolizing. They use oxygen and produce CO2 during their metabolism. Do you see over here this oxygen continuously coming in and this glucose coming in, right? The mitochondria will take these two, burn it, make ATP, which is needed for the cell, and they will form CO2 as waste and water, right? So this oxygen is needed to come in, and the CO2 waste will go out, okay? Alrighty. So, over here, right, before we do this, right, I want to go over here. And I want you to label each of these vessels as an artery or as a vein, okay? I'll give you 2 seconds. You can pause the video if you need. So remember what I told you, right? We bring deoxygenated blood to the lungs. We bring bad CO2 lungs to get rid of the CO2, to breathe it out. We breathe out carbon dioxide. So we are bringing this to the lungs. This is going away from the heart. Therefore, this is an artery, right? Then we are bringing back oxygenated blood to the heart. So this is coming back to the heart. This must be a vein, right? Then we get back to the heart, right? And we will shoot out this blood to all the tissues. This oxygenated blood goes to the tissues. So this is going away from the heart this way. So this must be an artery, right? And then at the tissues, it takes all the oxygen, puts its CO2 into the blood, and it returns back to the heart, right? So this must be a vein, right? Because it's revient, coming back to the heart. I want you to look at the 2 arteries here, this one and this one. Look at what color they are. So, as I emphasized so much before, does being an artery mean that you're oxygenated? Not necessarily. Look at the pulmonary circuit. The artery is actually deoxygenated, right? Look at the veins. Does being a vein mean that you're deoxygenated? Not necessarily. Look at the systemic loop is blue and deoxygenated, and look at the pulmonary loop, which has a vein that is oxygenated, right? So the definition of artery and vein means, are you going away from the heart or coming back to the heart? So that's our definition that we have. Okay. Now we're going to talk about the right side versus the left side of the heart. The heart is said to be a double pump that creates pressure to push blood through the circuits, right? Remember I gave you that analogy of a water balloon that you squeeze with a straw in it? Now our water balloon is getting a bit more complicated, okay? Our water balloon has like 2 straws on each end, and it has a divider in the middle. So this is a double pump because it creates the pressure to push blood, but there's this septum in the middle, okay? And this septum separates the heart into the left side and the right side, okay? And you're going to tell me, oh, sir, what's going on over here? You're telling me this is the left and the right? Remember, this is in the perspective of the patient, and you're going to have a lot of difficulties in the beginning, and you're going to get confused a lot. So what I suggest you do when you're doing a problem, right, is to actually write the, this is the patient's left, and this is the patient's right side, so that you don't forget, okay? You can write it on your sheet, or when you do a little doodle, put left and right inverted because it's the patient's perspective, right? So there's this septum in the middle that separates the heart into the left and the right side. And that means there's no mixing of blood between the two sides. Each contraction, though, is synchronized. It's a double pump in the sense that, yes, my water balloon has a division in the middle, but when I squeeze both sides, like my heart will squeeze together on each side equally, right? When this heart side squeezes, this side squeezes too. So it kind of squeezes on both sides, and blood simultaneously gets ejected from both sides into this side and into this side at the same time, right? When we squeeze it, there's like 2 straws on each side of the heart, and they will go into their respective straws and exit the heart out through their respective straws, right? So each contraction is synchronized and affect the 2 sides of the heart simultaneously. Okay, so I want you to look at the color and the actual arrows, right? The left side of the heart pumps oxygenated blood out over here. This tube goes in the back, so don't be deceived. Like, it's not switching sides. It's just that it goes out this side in the back of this vessel here. It goes out here, and it goes out. The left side pumps to the systemic circuit, out through the systemic circuit. And also, remember, there's a little coronary. There's a branch here that goes to the coronary circuit, okay? Do you see here the red? All these red vessels? Those are coming out from the aorta, actually. So they will go to the outside. So it's like a mini loop off of the systemic circuit that goes around the heart. But it's hard to show in this picture, so I prefer to show it here. So the left side of the heart pumps oxygenated blood out through the systemic circuit and the coronary circuit. And the right side of the heart, so all of this gets sent out to the systemic circuit. So again, let's practice a bit. This is a artery, part of the systemic circuit, because it's going away from the heart, goes to the capillaries, and then this becomes a vein because it's revient au cœur. And the right side of the heart, now do you see that it's blue? Why is it blue? Because now it's oxygen poor, right? All of these tissues, the little cells over here at this systemic circuit, it could be in the toes, could be in the head, could be in the arms. They took all that oxygen, and that turns the in our diagram, we put the color of the blood as this blue, right? Oxygen poor blood, CO2 rich blood. That deoxygenated blood with oxygen removed is like bad blood at this point. Now we're going back to the heart. And in the right side of the heart, we pump that deoxygenated blood on the other side, right? We're pumping now on this right side of the heart, right? This right side pump will send blood when it contracts up to the pulmonary circuit. Why does it send it to the pulmonary circuit? Because at the lungs, that's where we breathe in oxygen, right? When we breathe in oxygen, we can pick it up from our lungs and put it into the circuit. And look, now all of a sudden, my capillaries at the lungs are red. They are oxygenated. This is full of oxygen because I brought in oxygen from the lungs. And all this crap CO2 goes out and you breathe out that CO2, right? So you bring in oxygen into the blood and you breathe out oxygen out into the air. And now we have oxygenated blood that comes back on the left side of the heart, right? And it will get pumped again. Look, the left side has oxygenated blood because it went to the lungs. And it goes out through the systemic circuit and coronary circuit and gets pumped out to feed the cells of the heart, feed the cells of the body, the toes, the hair, right? And then it comes back with all that oxygen depleted and gone. And then it needs to go recharge at the pulmonary circuit, right? I just want to emphasize something. My favorite analogy to give is a road trip, okay? When you go on a road trip, right, you need to go and charge your car, right? So you don't go on a road trip with your battery being at 15%. You're going to go and drive a little bit to a charging station. You're going to go to the charging station. You're going to pick up and fill your battery of your car up, right? You're going to fill up your blood with oxygen because right now it's depleted, right? So you charge your car up to 100% and then you drive back quickly back home, right? It's not far, the charging station. You come back to your house, right? And then you pack your stuff up and then you do your world tour, right? Then you go do your long trip down to the toes or up to the hair, your far away trip, because now you're charged, right? You have oxygen, right? And that oxygen is going to get depleted over here. You're going to give away your oxygen as you do your world tour. And then you're going to come back with that lower battery. You're going to come back home, right? And eventually you're going to say, I want to do another trip, right? So you're going to go back to the charging station with that low battery. You're going to go charge back up. And then you're going to come back home and then you go back on your tour, right? So I like this analogy a lot because it kind of shows that the pulmonary circuit is to recharge, to refill with oxygen and get rid of CO2 waste, right? But the magic happens in the systemic circuit for sure. Okay. Now, there's this other little special circuit that's called the hepatic portal circuit. Anything hepatic, it pertains to the liver, okay? So hepatocytes are hepatic cells, liver cells. Hepatic portal circuit is a circuit that is for the liver. So I want you to know that the GI system has a unique venous drainage system, okay? So the gastrointestinal system has this venous system, meaning it's going away from the GI organs, and it drains in this interesting way. Over here in the diagram, you see the heart, the pulmonary artery, going to the lungs, deoxygenate, refill, bring back oxygenated blood to the heart. Then we have the systemic circuit, right? We have the aorta. It's coming off of the blood. This is the systemic arteries going out, going to other organs, coming back through the veins, and going back to the heart. But the GI organs have something interesting. When all of the aorta feeds all of these, right? Because the liver needs oxygenated blood, the stomach needs oxygenated blood, the intestines need oxygenated blood, but the hepatic system is interesting. Venous blood from the digestive system, right? Do you see this venous blood from the digestive system, from the stomach and from the intestines? They get sent through what we call this hepatic portal vein, and they will pass through the liver before going out into the circulation and back to the heart to get oxygenated. And the reason why the stomach and the intestine send their substances through this hepatic portal vein to the liver before, why couldn't they just go directly into the venous blood, right? It's for two reasons. Number one, nutrient processing. The intestines, right, they pick up all of this. These GI organs pick up all of the nutrients that we ingest, right? We ingest food, and then in our digestive organs, we bring all those nutrients and stuff into our bodies, right? So if we pass through the liver, actually the liver will help to convert some of the absorbed nutrients, the sugars, the amino acids, into usable forms or storage. Remember that sometimes we store glucose if we eat excess. Well, we pass right next to the liver, perfect. All that excess can go there. The most important one, however, is probably the detoxification. All of these things that we're absorbing are from the outside world, right? Remember, the digestive system is still the outside world. So any potential toxins that we ingest, right? Alcohol, let's say, or any other bacteria that might be ingested in, right, will get filtered in the liver. One of the functions of the liver is to filter out toxins and harmful metabolites, like ammonia, right, from the gut and prevent it from reaching the brain and other organs. So the liver is going to detoxify all of these toxins. So it's good that there's this passageway through the liver and then out afterwards, right? So we call this the hepatic portal vein because it's a portal between, usually you're just supposed to go into this circuit, but there's a portal into the liver and then out through the hepatic vein. So that's what's special about our liver, the hepatoportal circuit. So now we go back to our heart. This is that was just a little side note. What are the four chambers of the heart, right? The heart has upper chambers that are called the atria. And they have lower chambers called the ventricles. So look here. This is the right atria. This is the left atrium. This is the right ventricle. This is the left ventricle. Okay, these are chambers. Think of a chamber like something you could fill blood with, right? Blood fills up these regions. It's a chamber. Between each of atria and ventricle, right? The right atrium versus the right ventricle, and over here, the left atrium versus the left ventricle, are what we call the atrioventricular valves, or the cuspid valves. So I prefer the term atrioventricular because it says it in its name. It's a valve that's between the atrium and the ventricle, okay? So atrium, ventricle, there's a valve here. This is called the right AV valve, atrioventricular AV. So the right AV valve. This is called the left AV valve, between the left atrium and the left ventricle. That's why I prefer those names. The right side of the heart, right, and the left side of the heart, they do not mix because there is what we call, there's two septums or two septa There is the interatrial septum. Between the right atrium and the left atrium, there's this septum here that blocks the left atrium from talking to the right one. Here they're covered by all these vessels, but there's a septum between this atrium and this one. And then over here between the ventricles, these bigger chambers on the bottom, there is the interventricular septum. Between the ventricles. Between the ventricles, there's a septum here. And this is key. The right side of the heart never mixes with the left side of the heart unless it goes through the vessels. I want to go back over here. This is over here, your left side of the heart, right? It cannot cross into this side. The atria cannot cross into this side. It needs to be pumped through a circuit to go over here to get to the right side. Okay? Same thing over here. We cannot cross to this side. We cannot cross to that side. We need to be then pumped to this circuit to get to the other side. So the right side and the left side, they do not mix thanks to the septum in the middle. Septum, like your nose septum or a septum piercing, pierces through the septum, the division between the nose. It blocks the left and the right side of the nose. This is a septum between the left and the right side of the heart. What are the atria? The atria are the receiving chambers. The atria never mix because they're divided into the left atrium and the right atrium by the interatrial septum. Inter between the atria septum closure. Blood vessels enter into the atria, meaning that they are veins. Returning revient au cœur. Do you see over here this right atrium? You have these vessels entering, and actually this vessel over here from the bottom enters into the right atrium. I don't see any vessel going out of the atrium, right? Do you see the arrows here pulling into the right atrium? Here it's going into the right atrium. On this side, do you see these arrows here, 1 and 2, going into the left atrium? Revient au cœur, they are veins. So veins are returning blood to the atrium. That's why we say they are the receiving chambers. They receive blood. They do not expulse blood out of the heart. They receive blood from the 3 circuits, okay? They have thin walls over here. Look at the walls. They're very thin, these walls. Since they require minimal contraction to send blood. Look at the direction of the blood. From the right atrium, it sends blood down into the ventricle. This left atrium sends blood from here down into the ventricles. So they actually don't really need to contract that strong. And even the contraction they do contributes minimally to the propulsion of blood. Think of it. These top chambers squeeze down blood into the ventricles. But if you think of it logically, gravity will pull blood down from here into the ventricles. Gravity will pull blood down here into the ventricles. So I always call the atria a bit of a show off when they contract because gravity is pushing the blood down, right? They do squeeze the last drops of the lemon though, so that's okay. We'll let them do their thing. Atria receive blood from different areas. So what feeds the right atrium? Again, remember you're always going to mess up with the right and the left. So I know this is your right, but you're going to write left here. And this is your right, but you're your left, but you're going to write right over here, right? Even I'm confusing now. So what feeds the right atrium on this side? You have the superior vena cava. Superior because it's on top. This is the inferior vena cava on the bottom that feeds into here. Do you see this arrow? That's coming from the inferior vena cava. This is coming from the superior vena cava. The superior vena cava will bring blood, return blood from the body. Vena cava, V like vein, right? And so these are the big vessels that are returning blood from the heart. The superior vena cava brings blood from parts of the body above the diaphragm. Think of the head, right? Do you think that the returning blood coming down from the head will go all the way around and go to the inferior vena cava? No, that doesn't make sense. Your heart is here, right? So it'll go down and it'll drain actually instead into the superior vena cava and enter into the heart directly. Your neck, right? It will go directly into the superior vena cava. It's not going to go down and around, right? It's easier to just drain down through the superior vena cava. Your arms are technically above your heart, right? Your heart is down here. Your arms are technically above your heart. So all of that blood will drain into the superior vena cava. Same thing for things that are above your diaphragm. All of that will drain into the superior vena cava, okay? On the opposite end, right, the inferior vena cava returns blood from body parts below the diaphragm. So below your diaphragm, right, your legs, they will all work extremely hard to bring up the blood. Even there's valves. Remember the valves in the vena cava and the veins? They will bring blood back and they'll bring it back up to the right atrium. That's from the inferior vena cava. Your legs, right? Your pelvic area, your abdominal area, right? All of that below the diaphragm will be coming up through here. Makes no sense to go up this way, right? So I like to think of it this way. The superior vena cava brings back blood from the upper part of the body, right? And the inferior vena cava from the lower part of the body, right? And lastly, there's an annoying one, the coronary sinus, okay, returns blood from coronary circuit, right? When we feed the outside of the heart with arteries, there will be veins that pick up the CO2 and becomes deoxygenated. That needs to return back into the heart, right? Well, it's going to go into the deoxygenated section, right? So actually, you see over here the opening of the coronary sinus. This is where there's an outside vessel. I'm drawing it inside, but pretend it's external. That's going from the heart and returning into this the right atrium over here. So it returns blood from the coronary circuit from the heart tissue. That's going to be deoxygenated blood. The heart's going to, outside of the heart's going to pick up all the oxygen and push out the CO2 into that blood, and it'll come back blue, right, in this color. It's going to come back to the right atrium, and it will pool all here. So the right atrium really picks up all of this blood that is deoxygenated, right? What about the left atrium? The left atrium, it receives the left and the right pulmonary veins. These are the left pulmonary veins. The right pulmonary veins, you don't see them. They go to the lungs on this side. Let me show you here. So here are the left pulmonary veins that'll come from the lungs over here. And they will go over here and they will enter this way. And they kind of join in over here and they feed into the circuit over here and they enter in. You don't see them very well in this picture. So this is the right pulmonary veins on this side. I added them to the picture, pulmonary veins. Let me go see if the other picture has it better. No, it does not. Okay, we'll see a better picture soon. But something needs to bring those back, right? So the right pulmonary veins, we're going to send blood this way. And it transports, okay, let's get this through. Pulmonary veins. Pulmonary means it's coming from the lungs. Veins means it's coming back to the body. If it is coming back to the body, a vein, from the lungs, that means pulmonary, poumon, veins, revient au cœur. That means that it must be oxygenated, right? It must be red because it's coming back from the charging station, back from the lungs, and it's coming to our heart, right? So It's transported oxygenated blood from the lungs back to the heart. Why is there the left pulmonary veins and the right pulmonary veins? Because you have a left lung and you have a right lung, right? So the right lung, all those capillaries that went there, they will come back through the right pulmonary veins and enter into the left atrium. And the left pulmonary veins will come from the left lung and enter into here, this oxygenated blood. The atria, right? So this is what the atria receives blood from, right? Because remember, atria are the receiving chambers. Where is the atria releasing blood to? The right atrium, deoxygenated blood, will send its blood down to the right ventricle. Okay, the atria always send it to the ventricle. This left atrium sends blood to the left ventricle, so it puts it down into the bigger chamber, right? These atria are just the collecting chambers to receive all of that blood. What are ventricles? The ventricles are the discharging chambers. Discharge means to eject out, right? So the ventricles, they actually do not mix, and they're divided by this septum into the left ventricle and the right ventricle, okay? And this is the interventricular septum here that's dividing the left ventricle and the right ventricle. The ventricles make up most of the heart volume. They're the biggest part. These atria are tiny. They're tiny receiving chambers. The ventricle is the biggest part. Let me show you actually, I want to show you the real heart. Over here, right? These are the tiny atria over here. You don't even see it on this side. And all of this stuff here, right? These are the ventricles. It's crazy how small the atria are. Okay. So the ventricles make up most of the heart volume. The blood vessels that are, in this case, are exiting the ventricles. Do you see this right ventricle? The blood vessel that exits is this one. It's exiting. Look at where the arrows are going. And in this case, the left ventricle, look at where the arrow is going. It's exiting this vessel, right? So if blood vessels are exiting, they are considered arteries because they're going away from the heart, right? They are discharging blood into all three circuits. So the atria received blood from all three circuits. The ventricles will discharge blood out to the three circuits, right? The heart is just a pit stop to get repropulsed back into another circuit. The ventricles have a very thick wall. Do you see the size of this wall here, right? And this side of the wall is a bit smaller, but we'll talk about why that is. They have a very thick wall compared to the atria, right? Because they actually need to contract and send blood through the circuits, okay? This atria here needs to contract and send blood down here. That's nothing, right? This ventricle, however, needs to contract and propulse blood through this vessel all the way down to your toes and back. And it needs to propulse blood up through these up here to your head and back. So the contraction of this needs to be strong, or else you're not going to reach the proper distance. So the atria are thin-walled versus the ventricles are very thick-walled because they actually need to send blood through the circuit very far, okay? Ventricles receive blood from where? I already told you about this, right? The right ventricle over here receives blood from the right atrium. Do you see the atrium sending blood down? And the left ventricle over here receives blood from the left atrium. It's coming down over here, right? And then the ventricles squeeze and they send blood where? They send blood to different places. What exits the right ventricle will pump blood into the pulmonary trunk. This ventricle will pump blood into the pulmonary trunk here. which will then branch off. Do you see how it's branching here and it's branching here? It branches off and sends blood to the right pulmonary arteries and the left pulmonary arteries to go to the lungs for gas exchange. So uh, this this vessel over here, right, it's called the pulmonary trunk because it's the trunk of the tree. Do you see the branches here? It actually looks like a tree. So from here we go through the pulmonary trunk, and then we're going to go through the left pulmonary arteries, right pulmonary arteries. So this left pulmonary artery is going to go to the left poumon, left lung, and it's called an artery because it's going away from the heart. This side, we're also simultaneously going to send blood through this side to the right lung, poumon, artery, because it's going away from the heart, right? So the names actually make sense. Don't memorize the names. Try to make sense of them and it'll be a lot easier. What exits the left ventricle, right? So that was for the right ventricle. Now let's look at the left ventricle. So it receives from the left atrium, and then when it contracts, it goes through this big thing. This big thing is called the aorta, okay? The aorta sends blood into the systemic and the coronary circuits. So aorta starts with A, so it's an artery, okay? And you actually have two divisions of the aorta. It's the same tube, but this division here is called the ascending aorta up here because it's going up. And actually, this is called the arch, the aortic arch. This is the ascending aorta going up. And all of these little vessels are like little projections off. They will send blood to the arms and to the head and neck, right? Above the diaphragm. Remember that we had that division? Versus, and then you really don't see it in this picture, but this tube continues down in the back. I'm actually going to draw it down on the other side of this picture. If I was able to turn this picture around, this tube continues downwards and keeps going over here and becomes the descending aorta, right? Because this blood will go and feed the head, the neck, the arms, right? Whereas we still need to go feed the abdominal area, the stomach, the intestines, the legs, right? So over here, this descending aorta will go down and it will go to the lower extremities, okay? All right. So, now I went through the big vessels that are coming out of the heart. I need to now go into a bit of detail on the coronary arteries, okay? So the coronary arteries, right, are arteries, meaning they're going away from the heart, and they're going to the coronary, to the heart muscle itself, the external part of the heart. And they supply blood to the heart. There are two coronary arteries that arise from the base of the aorta. Do you see the aorta here is coming out of the ventricles? This is like the external view of the heart, and the aorta comes out of the heart, right? And it prongs, three little prongs here. This is a heart sliced open, so you actually see the aorta connected here, and there's your three prongs here. But let's pretend the outside of the heart is still in position, right? So there's actually two vessels that come off of the this aorta. There is the left coronary artery. This is the left coronary artery over here. It's on the left side. Let's go put our little left, our little right side. Okay. So you have the left coronary artery on this side, and that has two major branches. Over here, you have the left anterior descending artery because it's descending down to feed the heart over here. And then you have the circumflex artery. This is the circumflex artery. It does this circumflex loop-de-doop here, and it supplies the left atrium and the posterior wall of the left ventricle. So it's actually going to feed the the left atrium and the back wall of the left ventricle. It actually goes over here. On this side, you have the right coronary artery that then splits into two branches. You have the right marginal artery that supplies the right atrium up at the top and most of the right ventricle. And then you have the posterior interventricular artery that supplies the posterior ventricle walls in the back. Do you see this one over here versus this one, right? That's what we have going on here. So, okay, let's go through this craziness over here. Now I'm going to piece everything together for the circuits. Okay, this is a crazy diagram. It's really helpful, in my opinion. So, we're going to start at number 1. Okay, here's the vena cava. We're going to start at this number 1. I want you to look at the colors. I didn't put it in red and blue because, again, that mixes us up. I put the text in red and blue to know if it's oxygenated or deoxygenated, but then I put these little boxes to know if it's the pulmonary circuit, the systemic circuit, or the coronary circuit. Okay, so let's start at this point, this part of the diagram. This is called the vena cava. Number 1 over here or here. You could be superior vena cava or you could be inferior vena cava returning to the heart. This here is the heart, right? This is our heart. And then you have some vessels coming out of the heart, and some coming back to the heart, okay? So the vena cava enter into the heart, the right atrium, right? The vena cava is in purple because it's part of the systemic loop. It just came back from your toes or from your hair or from your arms, your legs, right? So the vena cava came back to the right atrium over here. Then we go down to the right ventricle over here. Then we go and we get the ventricle contracts and sends blood up here through the pulmonary trunk. This is the trunk here. And then to the pulmonary arteries, number 5. It goes either to the left pulmonary arteries to go to the left lung or to the right pulmonary arteries to go to the right lung. So the pulmonary arteries go to the lungs, right? From the lungs, we don't see this in this picture, but the lungs will take this deoxygenated blue. Do you see how it's blue? The pulmonary arteries has blue deoxygenated blood, takes the CO2 and breathes in oxygen and puts oxygen into the pulmonary veins, right? And so then from the lungs, those pulmonary veins will return right back to the heart. So 6 is the lungs will send out pulmonary veins. Veins revient au cœur. Arteries were coming out of the heart, going to the lungs. Veins were coming from the lungs back to the heart, right? So the pulmonary veins come back. There's the over here you have the left pulmonary veins, and over here you don't see them. There's the right pulmonary veins from the right lung. And they go back to the left atrium. So we go back to 7 here, the left atrium. And then we will go from the left atrium back down to the left ventricle, right? Left ventricle 8. And then all of this was the pulmonary circuit going to the lungs. This purple is the systemic circuit. So from the left ventricle, we squeeze the left ventricle hard, and that will go up here to number 9, the aorta. Either to the aorta up to the head to feed our head and neck and arms, or it'll go and veer downwards over here. I should have actually put a 9 over here. And this will go to the descending aorta to feed our stomach, our legs, or anything below, right? So that is the aorta. And then it'll branch into little systemic arteries, right? Aorta, arteries, arterioles, and then we go and go to the peripheral tissue. Whatever that tissue is, here's the little tissue tower. Maybe it's your toes. Maybe it's your stomach. maybe it's your hair, right? Maybe it's your arms. Wherever that is, it goes to that tissue. And this, do you see again, once again, this is the place where the red text becomes blue, because the systemic arteries will give its oxygen to the peripheral tissues, right? And the peripheral tissues will give its junk and CO2 to the systemic veins, right? So in the peripheral capillaries, we take the oxygen, we remove it from the blood, and we put CO2 into the blood. So we have this deoxygenated blood. The systemic veins come back to back through the vena cava, number 1, right? And we return back to the right atrium, number 2. And on and on and on. This goes and loop-de-doops over and over. So forget all of this orange stuff here, right? And just look at how there's a pulmonary loop and there's the systemic loop. On top of these loops is a mini systemic loop. That's why it's up here near this systemic loop, because there's a loop for the coronary circuit. From the aorta, let me go back over here. From the aorta, where was it? The coronary. Yes. From the aorta, remember there's these 2 branches coming out. It doesn't go up here. There's 2 branches that come out here. Those are the coronary arteries, right? So from the aorta, they go to the red vessels. The coronary arteries are oxygenated, ready to feed the outside of the heart. It goes to the heart muscles, and the heart muscle will take in the oxygen, right? And it'll put out the CO2 into those vessels. And that becomes coronary veins, deoxygenated. And that will return back to the right atrium via the coronary sinus. Remember this little hole over here, the coronary sinus? That will bring back the vessels from outside the heart and come back to the right atrium, and then it goes to the lungs to get reoxygenated, right? So this is just a mini parallel loop for the heart, right? But it's the same principle as the systemic circuit. Very important thing. The left ventricle, if you look at the heart, the left ventricle is thicker than the right ventricle. This wall here in the picture is thicker compared to this wall here. Remember, this is right, this is left. If you look at a cross section, look at the left ventricle, how thick it is. Look at the right ventricle, how thin it is. Okay? The right ventricle pumps blood where? Let's go over here. Actually, let's go to a nicer picture like this. The left ventricle pumps blood up to the lungs. The right ventricle pumps blood to the systemic circuit. Okay? That's the key over here to understanding this. So the right ventricle pumps blood through the pulmonary circuit, les poumons, the lungs. Are the lungs very far from the heart? No. They're like right next to the heart, right? So the contraction is not needed to be super strong. There's less contraction needed, so there's a smaller wall of the heart here. The left ventricle, however, pumps blood through the systemic circuit. Some blood needs to get to the toes and back to the heart. Some blood needs to get to the tippy top of the hair and back down. So the worst one is the toes. Look how far the toes are, right? So this blood that's contracted here needs to go up through the aorta all the way down to the toes and back. So the contraction needs to be much smaller. This blood here goes through the pulmonary trunk and needs to just go to the lungs and then back, right? So that's why this left side is thicker than this right side over here. All right, so let's tackle this problem over here. We're going to label these things first. So the first thing we have is the aorta to label. The aorta is quite easy to find because it is what is carrying oxygenated blood to all of our tissues in the body. So it needs to go up to feed our hair and our head and our neck and our arms, and it has to go down to feed our legs, right? So do you see this vessel here with the three little spikies at the top, right? Those vessels are feeding our head, neck, and arms, right? And then down here, 10, everything below the diaphragm is being fed by this, right? So all our abdominal organs, our legs. So this 10, this entire 10 is the aorta, and it starts from here and it goes, you don't see it connected here, but it goes behind and it goes up, and this goes up to our head, neck, and arms, and then this will go down, right, to the capillaries over here that it's trying to symbolize. So aorta is 10, okay? Our lungs are 9. These are trying to symbolize our two lungs, so this will be 9. Our left ventricle, right? Remember, I should have done this in the beginning. This is the patient's left, and this is the patient's right side. So the left ventricle, the ventricles are always the bigger chambers on the bottom. They should be bigger actually in this diagram, but this is not accurate in terms of size. So I would say that the left ventricle is 4. The pulmonary artery. Pulmonary means it is going to the poumons, the lungs. Artery means it's going away from the heart to the lungs, right? So what is going to the lungs? We have a few options here. So here are the lungs, and I see that there's really two vessels connecting. There's one that's coming over here from this ventricle, and there's one over here, right, on this side that I see coming to the atria, right? These two are coming to the atria. These are going over here. So, first thing, if I am ejecting blood out of the heart, exiting the heart, right, the arteries, what chambers are ejecting blood out? Is it the atria or is it the ventricles? It's the ventricles, right? So the ventricle here will be ejecting blood out and sending it to the lungs, right? Actually, this is called the pulmonary trunk, and then it splits into the two pulmonary arteries, one for the right lung and one for the left lung. So I know that I definitely know that the blood is going in this direction here, even for the aorta, right? I know blood is going in this direction because anything that is exiting the ventricles is getting sent out of the heart. Remember, the atria are the receiving chambers. So actually, I'll draw it here. Atria are the receiving chambers. They're receiving blood. Here, it's over here, right? They're receiving blood. And the ventricles are ejecting. So you see that. So it makes a lot of sense that this over here, the pulmonary trunk, and then here, the two pulmonary arteries going to the lungs. So 7 is going to be my pulmonary arteries. The right atria are on top. That would be 1 over here. The capillaries, I see a little capillary bed over here, right? It could be any capillary. Remember, there's many capillary beds, but here we're just showing one to go to tissues. Pretend that this is maybe the toes that we're going to feed, right? The cells of the toes. And then the pulmonary vein. Vein revient au cœur, right? It's coming back to the heart, and it's pulmonary. So whatever from the lungs is coming back to the heart. And remember, we said that these two here, right, it's coming back to the atria. So 8 would be our pulmonary veins. The left atrium, 3. Vena cava, vena, it has a V, so it's a vein, right? It's coming back to the heart, revient. And it's the big ones that are bringing back blood to the heart. So remember, there's a superior vena cava and inferior. And here you see that, let's say the head or the arms over here, this is the superior one bringing blood back, right? So that's part of the vena cava. And this fifth one, if all the blood coming from our toes or our abdominal organs, that'll come up through the inferior vena cava. So 5. And then lastly, the right ventricle. So here's the right ventricle, 2. That'll be what we have here. Remember that arteries carry blood away from the heart and veins carry blood towards the heart, right? The aorta is an artery, right? Actually, I'll just write A over here. let's just write A for artery. The pulmonary artery, this is an artery, right? Then the pulmonary vein, that's a vein. And the vena cava, that's a vein. Everything else is just like pieces to the puzzle, right? But these are the 4 big vessels that come out of the heart. I want you to go and draw where there's O2 rich blood or O2 poor blood, right? This is also known as, this O2 poor blood is also referred to as CO2 rich blood, because if it's lacking oxygen, it gave away all its oxygen, probably picked up all the CO2, right? And this blood here is actually CO2 poor. It's full of oxygen, but it doesn't have much CO2, right? So another name for this is, we call it oxygenated for red, or deoxygenated for the blue, oxygen poor blood. So let's go draw where all our oxygenated blood is. Remember that the left side of the heart has all of this oxygenated blood, and that the right side of the heart has all this deoxygenated blood. That is the premise of our double pump of the heart, right? That there's really a split thanks to this septum in the middle. And where are we pumping blood to? We are pumping it through the aorta. So all over here, right? All of this. And then we're going to oxygenate up here, up here, up here. And then down and around. And then to the capillaries, right? We're going to go to these capillaries here. Where else are we going to go? We will also go, where did that oxygenated blood come from? It came from the lungs. The lungs put oxygen into the blood. So let's go backwards from our heart, right? We did all of this circuit frontwards. Now let's go backwards from the heart. Where did it come from? It came from here, from the lungs that oxygenated the blood, right? And it came from here, right? This one, where this lung oxygenated the blood and brought it in. Now let's go and note where all the deoxygenated blood is, right? Let's start from the heart. Where is it going to send out? So blood comes over here in the capillaries. The cells in the toes will steal all of that oxygen and make it oxygen poor blood, right? So you're going to see here that we have all this poor oxygen blood because all this oxygen was given to the cells, right? Here there's some toe cells that'll steal all of that oxygen, right? Because it needs it. And then it puts a bunch of CO2 in here. And so that will come back through the vena cava back to the heart. And if it's our hair cells on the top, that's the capillary beds that are here, then that blood will come down this way instead, right? There are some cells here that will steal up at the top of your head and neck and arms that'll steal some oxygen too. So that's going to come back down. And so that's where it's coming from. It enters the heart, and then where does it go? That blood needs to be sent to get reoxygenated, right? We just came back from our world tour, and now we need to go and back to the charging station, back to the lungs to fill it back up. So we'll go up the pulmonary trunk, and some of it will go to the left lung, some of it will go to the right lung. And in the lungs, then we can inhale oxygen in and put it into the blood. And then it'll come back out over here through our pulmonary veins. That'll come back to the heart. Last thing to note, arteries have relatively high pressure, veins have relatively low pressure, right? So all of these arteries that are being, remember what an artery is, it's going away from the heart. So here's the ventricle pumping out through the aorta. Here is the ventricle pumping the pulmonary trunk to the lungs. Those are high pressure, they're closest to the heart, right? The lower pressure, right? Think of the aorta. This is high pressure, high pressure. Then it gets all the way here, and by the time it's at the end of the garden hose here, it's dribbling back and it's trying to counter gravity. The pressure is very low. It's actually, there's little valves here that are trying to get it back up to the heart, right? So arteries are relatively high pressure, veins relatively low. All right, so now that we've went through the heart chambers and the vessels, I'm going to add a layer of difficulty. I'm going to add valves, because maybe you've thought about it before, but why is it that blood decides to only go in one direction? Why does the blood go only through the arteries and it doesn't go up through the veins, right? And it's thanks to valves, right? Over here, these are some valves we'll talk about. There's one valve here. These are our atrioventricular valves. And then you have some valves over here. These are the cuspid valves. These are your semilunar valves that we'll talk about. So, what are the 4 heart valves? Valves are, they are like doors. They're one-way opening doors, right? These are doors that only open one way, and you cannot open it the other direction, right? So you know those doors that you can open them this way, but they cannot be opened this way, right? It's only this way that you can open it. That's what a valve is. And valves are always between regions, and I want us to separate those regions. So first of all, we have the atrioventricular valves, or some people call them the cuspid valves. And it says it in their name. I love this name because it says atrioventricular. It separates the atria from the ventricles. So just go locate this atria and this atria, and you will find the valve. This is the right AV valve over here. Okay, do you see this flap here? And the little strings attached? That's the valve. The little sheets, they close, and they will prevent blood from regurgitating back. Look at my animation here. Do you see these AV valves? When the atria contract, it pushes, it allows blood, but then the valve closes, right? So this is one of the valves, the right AV valve. This one is called the left AV valve. AV for atrioventricular. So one thing to note, the right AV valve could also be called the tricuspid valve, because you don't see it very well in this picture, but there's 1, 2, 3 little flaps. You know the parachutes, right? So over here, let's go back. This has 3 little parts to it, whereas this one only has 2. Do you see the strings? I see 3 little strings to my parachute. And then this one here, I have 1, 2. So the right AV valve is called the tricuspid because it has 3 cusps. It has 3 little flaps, whereas the left AV valve is called the bicuspid valve because it has 2 little flaps, right? Think of the flaps as they hit together and they block blood from entering back up here, right? Blood won't pass back because they're closed. And then they open up and then they close. So this is your right AV valve or your tricuspid. This is the left AV valve or bicuspid. In medicine, we also often use the term mitral valve. So I know it's annoying to know all these names because they all mean the same thing, but sometimes in some papers or in some references, some professionals will prefer to use mitral, some will prefer AV valve, some will prefer bicuspid. So I need you to know all the names regardless, right? If you just memorize that, like, those names are associated to left AV valve, you're fine, right? I personally like right AV and left AV because it makes sense in terms of where it's located, but just know it's equivalent as well. Then we have the semilunar valves. They separate the ventricles and the major arteries. Remember I told you a valve always separates 2 different locations, just like a door always separates 2 locations, like the bathroom and the kitchen, or the bedroom and this living room, right? So over here, the AV valve's easy between the atria and the ventricles. The semilunar valves, right, they separate the ventricles and the major arteries. So here's a ventricle, and on this side, here's the pulmonary trunk, a major artery, right? And so this is our semilunar valve. It's called a semilunar valve because it has like a semi means half, lunar, moon, une demi-lune. It has this half moon, right? do you see in our animation here, you see that this valve is like a little cusp? And did you see it open, right? Look at the animation. The little moons, they open up and then they close back. They open and they close back. So that's the semilunar valves here. They separate the right ventricle from the pulmonary trunk. So there's this semilunar valve, and then over here is a ventricle, and here is the aorta, right? And unfortunately, this drawing is really bad. You don't see it, but here, there's the same semilunar valve that's over here. It's between this left ventricle and the aorta. It's right over here. It's hidden, but there is a semilunar valve, I promise. You see it a little bit over here. Do you see these little semilunes, right? The half moons. That is your other semilunar valve, and it is controlling the blood flow between the ventricle and this aorta over here. Okay, so again, I told you where the semilunars are. Let's differentiate them. This one here is called the pulmonary semilunar because it is between this right ventricle and the pulmonary trunk, which is going to go to the poumon, to the lungs, right? So it is in between it's the one that's leading to the pulmonary route to the lungs. So it separates the right ventricle and the pulmonary trunk. The aortic semilunar valve, right, is a valve that separates the left ventricle from the aorta, right? Aortic, aorta, not artery, aorta, right? Aortic. So this is separates the aorta from this left ventricle. If you slice the heart in half, right over here, you will see this, right? From up top, you will see this. If this diagram confuses you a bit, don't worry about it too much, okay? But I want you to kind of understand that these are doors, right? These are doors either between the atrium and ventricles, or these are doors between the vessels and the ventricles, right? So over here, you see your semilunar valves, right? Those would be this one and the aorta. That's why they're like next to each other here, because like this is sliced in half, right? I just sliced the aorta off, but if I push through these doors here, I would see this ventricle. And if I push through these doors, the pulmonary one, I would see this room over here. And then on each side, I have my tricuspid. These are like parachute sheets, or bed sheets, right? They're like big parachutes that will open and close. So here I have my tricuspid one. That's my right AV, and this is my mitral one. It's as if we kind of chopped off the atria, right? If I push through these doors, I would also see the ventricles and the right and the left ventricle. So how do valves open and close? The heart contractions force blood through the valves, okay? The valves work passively by responding to pressure differences on each side of the valve. So what I want you to know is that liquid is not compressible. A gas can compress, right? You can take a balloon and start to squeeze it and it'll kind of move around. Over here, you have liquid, right? Like blood. And you see that when you press one end of the syringe, it'll force the other one to move. So liquid can generate a pressure, right? Blood can generate a pressure, blood pressure. So how the heart valves work is very passively by responding to the pressure difference. People think that valves, they are a muscle that close and open, right? But that's not true. Valves, I want you to think of them like parachutes or bed sheets. Parachutes do not have a motor in it to open them up, right? parachutes, when you jump out the airplane, they will open up because there's a pressure, right, of the air that pushes it on one direction, and there's less pressure on the top. So you actually get the parachute to open up that way. There's no motor in the parachute sheet, right? So I want you to think of it, valves this way. Valves do not contract. I don't like that word. Valves don't contract. They open up passively, right? When blood, now instead of air in the parachute, I want you to think of blood, liquid. Imagine I had a big parachute sheet and I took an immense amount of blood or water and I threw it against the parachute. The parachute would open up as well, right? It's just the same concept as air hitting the parachute. So how does the heart prevent backflow, right? The heart valves allow for blood to flow in one direction. So here is my heart, it's empty. But I need you to know that there is blood here. And I want you to imagine what the blood would do to close these. So here's my AV valve, right? Do you see my two AV valves? This one's nicer to see. So when the heart so there's the atria and the ventricles, right? When the atria contracts, okay, the atria at the top here, that will open up these valves, right? I want us to imagine, actually, let's look at this side. Here's my atria and here's my ventricle. When my atria contracts, right, this generates a big pressure buildup here, and that can open up the valve. It'll create a pressure. I want you to think of the atria as a water balloon, and you squeeze it, right? It'll push against this valve, and that valve will open up, right? The bed sheets eventually, these like 3 little sheets will go and open up because the blood will push against them, right? That's when the atria contract and the ventricle are relaxed, okay? Now all of a sudden, that's the first beat of the heart, right? So the heart, remember, it does this weird beating. Do you see how the atria contract and then the ventricles contract, right? We have this kind of off sync of the heart. So when the atria contract, it pushes blood into the ventricles, right? That opens up the valve. Then all of a sudden the atria will relax, and now it's the ventricles' turn to pump. The ventricle will start to squeeze the blood, but interestingly, the blood, when the ventricle starts to squeeze, it pushes against the little parachutes here, but in the opposite direction. And remember I told you that this door only opens in one way? This door only opens this way, right? Let me show you over here. This door could only open up in a direction that is inside, so going inwards, right? So now they're open, right? And over here, it's not like these little parachutes can open up in the other direction, right? They can only open up downwards. So actually, when blood hits the valves, right? When blood eventually will start to hit on these over here, and they close together. So I actually like to show it with my hands, right? So when the atria, they contract, right? Up here, they start contracting. It pushes on these, and they open up, right? And then eventually the atria will stop contracting, and instead the ventricles will contract. And when the ventricles contract, right, here were my open up AV valves. When the ventricles contract, right, the blood will push against here, and they will close the AV valves, right? So over here, you see that heart valves allow blood to only flow in one direction, right? So it went from the atria down, and then when it gets sent up into the arteries, this AV valve, when the ventricle contracts, it closes the AV valve. So instead, the blood can go up here into this open space where the aorta or the pulmonary trunk is, right? In through the artery, right? So there's this direction, right? And then there's upwards, and the valves help to control that. So these are flaps, right? These are going to be the flaps. I want to just go through one thing before we explain it more in depth. How do the valves open and close? The AV valves over here are your AV valves, right? They are attached to the chordae tendineae. The AV valves are really just these flaps here. Do you see these little, I like to call them my parachute or my or bed sheets, right? They are just these big flappy like sheets, right? And they are actually attached to what we call chordae tendineae. Do you see these little strings here? I really like the parachute analogy because parachutes also have strings, right? So here's our valve, and then here you see chordae tendineae over here, right? Anchored to the papillary muscle. This is the papillary muscle over here. This is a little muscle that anchors it down to the ventricle wall. So I'll show you over here. Here's the strings. These are the chordae tendineae, and here are the papillary muscles at the bottom. So if you look over here, right, you see the little cords, and during ventricular contraction, when the ventricles contract, right, and the valves close, right, these little valves here, they will close. This valve here closes, right? The papillary muscle and the chordae tendineae actually keep the cusps from inverting into the atrium. The papillary muscle, it is contracting, but it's not closing the valve. I want you to forget that the closing of the valves only happens because blood is gushing to it. Actually, the papillary muscle and chordae tendineae, they prevent inversion of the valve. Do you see over here? Imagine your parachute had no strings, right? And you just threw the sheet out. Well, actually, it wouldn't do this nice round because there's nothing holding down, right? Or if you chop the cords, your parachute wouldn't work anymore and it would lift up. It's the fact that these little cords are holding it down that the parachute is not inverting, right? So these chordae tendineae and the muscles, they're holding down the parachute just to prevent it from, like, remember I told you these valves, right? What they're doing, they're like this, and they open and then they close. They open and then they close, right? They open and then they close. And it's if these valves, right, they never close and then open up this way, right? They never open upwards. They only do this kind of a movement because at this point here, the little chordae tendineae are pulling to prevent it from inverting backwards, right? So it's good that it's only opening in one direction. That's our one-way door. The chordae tendineae are preventing them from inverting upwards, right? Just how the parachute, the strings are preventing it from inverting upwards. So that was for the AV valves. The semilunar valves, they're actually only composed of 3 cusps. So over here, right, you see that this is one of the semilunar valves, and it has 3 little semi-lunes, right, moons, right? And then over here, you have 3 also. Do you see how this one is open, this semilunar? You see the open kind of holes that allow blood to go through. Here they're closed, right? So you see that this is just showing how they kind of function. They do not have chordae tendineae or papillary muscles, and backflow is less of an issue due to the high pressure in the ventricles contraction. So we don't have this same issue as the atria and the ventricles, okay? So the semilunars, it's interesting too. Let's go back to our picture here. I want you to think of the semilunars. These are little semilunes like this, and they open upwards and they close. They open and they close like this, okay? So that's how they work. You see it working over here, and the other semilunar, you don't see it because the aorta entry is hidden behind here. How do valves open and close? This is a key part to understanding everything together, okay? and you really need to know your anatomy well to move on to this part. I need you to know that when the heart pumps, okay, the atria squeeze to squeeze out all of the blood that they received, squeeze it all out into the ventricle. The ventricle is relaxed. So when the atria is contracted, the ventricle is relaxed and it's receiving all of the blood and filling up, right? This is the big chamber filling up with blood. Then, part two, the atria relax and the ventricles squeeze to shoot blood out of the arteries, eject it out of the heart into the circulation, right? So we're going to split this up in two parts. When the heart, or I should actually have said, when the ventricles are filling up, filling the ventricles, right? It's still filling the heart. And part two is emptying the heart. So first thing I want you to note, okay, when we're filling up the heart, we have the atria contracted. Do you see that here the atria is contracted, right? There's arrows here. We have all this blood that's in the atria, and we are contracting it, squeezing it down into the ventricles. This happens on the right side of the heart and the left side at the same time, right? The two atrias are squeezing into their respective ventricles. So this should make sense that when the heart is filling, this is squeezed and the ventricle is relaxed. It's picking up all the blood as much as possible. If the ventricle was contracted at the same time, they'd be fighting, right? The two would be squeezing and pushing against each other. So they actually take turns and the heart goes like this. Boom, 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 boom. Okay, so filling the heart, we're like this. Squeezed atria, ventricle, relaxed. When we're emptying the heart out, right? Do you see the blood being ejected out of the vessel? The ventricle is squeezed, contracted, and the atria is relaxed, okay? So that's why we see this pattern here. So that's emptying the heart out, releasing it. We have a relaxed atria, right? And here, do you see these arrows that are showing a squeezed ventricle? Look at the size of the ventricle too, right? Compare this versus over here, right? Here it's opened, it's a big space, allowing to collect, right? And then here we squeeze the ventricle. Here the atria is relaxed and open, and over here it's squeezed, right? So I'm going to go through these two phases. There's very different things happening to the valve in each phase. When we're filling the heart, the atria is squeezing. It's collecting blood into the ventricle. What happens to the AV valve? Okay. Blood that was received by the atrium flows down to the ventricle, right? We're filling up the ventricle, and the AV valve, right? Or cuspid valves, I don't like that name. Let's think AV, it's between the atrium and ventricle, is going to be open. I just want you to think of this logically. Why would the AV valve be open? Makes sense. We want to send blood down to the ventricle, so we need this door to be open, right? So the blood will pool downwards. We need to open this, right? And remember I told you that the reason that valve's open is not because a muscle is pulling it. It's not because, opening and closing valves is not because anything mechanically is pulling it, right? It's simply due to blood being higher in blood pressure being higher in one region versus another. And just like the air in the parachute, the air pressure that pushes up on the parachute is higher underneath it than on top of the parachute. So when the atria is squeezing, right, the atrial pressure is bigger than the ventricular pressure. I want you right now to go and draw arrows, okay? I want you to go in the atrium and think of you being a little red blood cell over here. A little red blood cell in the atria, right? And getting squeezed. The pressure you feel around you is huge, right? So this pressure here, I want you to draw many arrows pushing against here. This atrial pressure is huge, right? Since the atria is squeezing. And over here in the atrium, right? I want you to imagine being in this relaxed ventricle that's not squeezing at all. The blood is going to start pooling over here, right? And accumulating. this pressure here is very weak. I'm going to put one arrow to counter it, right? But the atrial pressure is much bigger than the ventricular pressure, okay? Now, that is for the AV valve only. When you want to learn about and think about the pressures, I need you to first select the valve of your choice. So right now I'm saying the AV valve. And then ask yourself what two regions are between that valve, the atrium and the ventricle. And at this moment, what pressure is higher, in the atrium or the ventricle? Well, in this moment, when I'm filling the heart up, the atrium is squeezing and the ventricle is relaxed. So this pressure is bigger than this one, therefore this valve must be opened, okay? Now, I'm actually going to skip the semilunar for now. I want to go to emptying the heart out, right? Remember, this is when my ventricles will contract. Do you see this ventricle contracting? And the atria is relaxed. Remember, they go in this cycle like this. So right now, at this moment, this AV valve is going to be closed, okay? Logically, why is it closed? Because I actually want to send, when my ventricles contract, I want to send blood up this way, and I do not want it to enter back into the atria, right? So I do not want it to go back. And so what you need to do for the AV valve, right? The AV valves are closed since what? Since the ventricular pressure is bigger than the atrial one. Here I want you to go draw your arrows again. Since my ventricle is now contracting, it's the ventricle's turn to contract when you empty the heart out. Well, it'll push against over here, this AV valve, right? And the ventricular pressure is big versus the atria is relaxed, right? This pressure is very small over here. So this pressure would win and push against the little leaflets here, the little parachutes, and close them up. So that's for the AV valve when it's emptying. So when we are filling our heart up, it's very good that we are actually opening these doors to let blood pool down. And when we are when we are emptying the heart, we don't empty the blood back into the atria. We want to empty it into here. So it's good that this door closes so that it could be shunted to this direction, okay? So now I want to move on to the semilunar valves. And we're going to do the same exercise. What you need to do is ask yourself between what regions is this valve? And in this moment, where is my pressure higher? In this region or this region? Okay, so the semilunar valves, what locations are they between? They are between over here, the artery, right? This is the artery. And over here, they are between the ventricles. This is called the ventricle, right? Your arterial pressure, your blood pressure in your vessels, right? Like for example, when you measure your blood pressure with a machine, that pressure is really high, okay? So this artery pressure, this arterial one, specifically the aorta here, this is an extremely high pressure at baseline, okay? When we're just relaxed, this is a very high pressure that's in the arteries. So I'm going to put many arrows here, right? And the semilunar valves, they're closed since the arterial pressure over here is much bigger than the pressure going against it on the other side, the ventricular pressure. When you're filling the heart, the ventricle is relaxed, right? So the ventricle is relaxed, meaning that this pressure wins over and opens this valve, but this ventricle is relaxed and means that this pressure here is very low and doesn't beat this pressure in the artery. One thing about this drawing that's confusing, it's not actually the drawing, it's just kind of the concept that the AV valve, remember I told you valves are one-way doors. The AV valve opens up like this and it closes. Opens and closes. It never goes upwards, right? It just goes like this. So you see it here, it will just open and close that way. It will never go this way. The door doesn't open in the other direction. This valve here, the semilunars, they're opposite. They're little cusps like this, and they open this way, like this, okay? They never go invert like that, right? They only open upwards. So when this is pushed, right, there's a big pressure here, the doors are closed and they're never going to open downwards, right? They just don't open downwards. So that's a little bit confusing in this diagram. But so this arterial pressure is much bigger than this ventricular pressure here, okay? When we are filling the heart. Because when we're filling the heart and the atria are squeezing, the ventricles are relaxed and this pressure is not going to beat this pressure. So when we're observing the valves, right, you always need to ask what is between each valve. Like over here, we thought about the pressures for the AV valve. We thought about the pressure here versus here. Whereas the semilunar, we thought about the pressure here versus over here, okay? Now, when the heart is emptying, okay, your ventricle here is going to squeeze, right? And all of a sudden, this pressure, when you start squeezing your ventricle, this pressure starts becoming a lot, right? I told you that this pressure was pretty high, right? But eventually, when the ventricle squeezes, this pressure beats the pressure on this end, right? And it bursts the doors open, right? It pushes and bursts the doors open. So the semilunar valves, right, they're open since the ventricular pressure beat is bigger than the arterial pressure in the arteries. This is the artery, right? So we actually beat the arterial pressure and boom, we open the door up, right? So like piecing this all together, it's very cool because in this moment, right, when we're filling the heart, we're making sure that blood goes down to the ventricle and that, you know, the blood that's in the artery isn't coming back into the ventricle because the doors are closed, right? But then when we contract the ventricle, right, and we squeeze, then we're making sure that blood goes out this way into the artery, but it does not go back up into the atria and back into these vessels over here, right? That's what valves do. Valves route things in a specific direction and only allow flow in one direction, okay? So if you like these pictures, look at them, go for it. Remember I told you AV valves are flappy sheets, right? And semilunar valves are the little semilunes, right? The little cusps, I wouldn't say cusps because that's the AV valves, little half moons, right? Little semilunes. Why I do like this picture is this only showed one side of the heart, right? But you do know that this happens on the other side. There's an atrium over here and a ventricle here, right? So actually, you know, everything we talked about here happens on the other side of the heart too. So do you see how here we said that the AV valves are open when we're filling the heart? This is the AV valve over here that's filling up the ventricle. So blood will go down, right? This is like a heart sliced in two, so blood will go down. Same thing on the other side. Blood will go down into this ventricle. And these semilunar valves are closed, right? This semilunar valve is closed and this one would be closed too. Then when the heart is emptying, right, we're going to close these AV valves. So this AV valve is closed. This AV valve is closed, preventing the blood from going back into the atria. And at the same time, right, these two semilunar valves here are going to be open. So all the blood that's in the ventricle will exit out of here, or on the other side of the heart, they will exit out from this one, right? So here I wrote the right ventricle pumps blood through the pulmonary circuit, and the left ventricle pumps blood through the systemic and the coronary circuit. So in other words, the aorta, and over here, the pulmonary trunk and pulmonary arteries, right? So these are our open valves. So here you see actually a real open semilunar valve. here you see the semilunar valve closed, right? All right, so how do valves open and close? I think this is to just summarize everything we talked about. And I want you to stare at this animation for a bit, okay? Look at how when the atria contract, the ventricle relaxed, right? And then soon after the atria contracting, then the ventricle will go, right? It kind of looks like this, right? So we contract our atria, blood goes down, right? And then once we contract our ventricle, this one over here opens, right? So I want you to see it in two parts. Squeeze, send blood down, right? And then this one should be closed at that moment, the semilunar. Part two is we squeeze the ventricles, we close the AV valve, and we send blood and we open this door so that we can go up the artery here, right? So those are your two phases. So I'm going to read these to make sure that this makes sense. So the blood collects in the atria, right, over here. No valves needed for that. Blood just pools back. You increase the pressure in the atria, the increasing pressure in the atria, because it'll eventually start to squeeze, right? Forces the cuspid AV valves to open. So these valves are forced open, causing the ventricles to fill up, right? Then the blood expands the ventricles, right? And it starts closing the AV valves because the ventricles will start to contract and mount the pressure, right, in the ventricles as it rises. And then eventually the pressure will mount so much that over here the semilunar valves will open and burst open, right? And so blood is pushed into the arterial trunks, either the pulmonary trunk or the aorta on the other side. And as the ventricles then, we squeeze, as the ventricles relaxed, right, the blood pressure drops and the semilunar valves will close, right? Eventually the ventricle relaxes back, and this pressure here will beat the pressure in the ventricles, and so this semilunar will close back. And then eventually the cuspid or AV valves, they will open again, right? This valve here will open again, and the ventricles will begin to fill again, okay? So, that's for the heart valves. Next thing is the conduction and the contraction of the heart. So we're gonna revisit this, right? I know this was a lot, but we're gonna revisit it. I just want to take a break, and I want to go through the electrical activity of the heart, and then we're going to piece it all together, okay? So let's start with the conduction and contraction of the heart. We talked about this heart that pumps like this, right? Well, how is it that it's pumping, right? We know that it's cardiac muscle, but how is this cardiac muscle? Cardiac muscle doesn't contract on its own. If you put cardiac muscle on a table, it would do nothing. You need to give it a signal to contract, okay? And so there's actually, what is it that allows the cells to have this conductivity, right? First of all, we have these contractile cardiac muscle cells. These are the ones I was talking about. 99% of the heart cells in the atria and ventricle are what we call contractile cardiac muscle cells, okay? So they will depolarize and cause contraction, right? So something will stimulate them and they will contract, right? And so their function is really just to contract and to pump blood, okay? So if you look at the wall of the heart, right, especially that myocardium, you will see all of these cardiac contractile cells. These are just cardiac muscle cells, right? But I'm emphasizing contractile because these are the ones that are going in and out and contracting the heart and squeezing it, right? So these are the ones, these are our contractile cardiac cells. Remember that they have little gap junctions in between so that we can talk to the next cardiac cell and help it stimulate, right? So there's communication between the cells. Well, those are our contractile cardiac muscle cells. They contract, but as I said, if you put it on a table and wanted it to contract, it would not do so on its own. The reason why these cells contract is because these electrical cells, which are a separate thing, they actually will control and coordinate the heartbeat. They will establish a heartbeat, right? We say that they're responsible for the heart's autorhythmicity. They are responsible for the heart's auto means on its own, autonomous, rhythmicity, it's autonomous rhythm. And in this case, there are really two sets of electrical cells. Do you see how I split it? There's muscle cells, right? Like the actual cardiac muscle that are kind of dumb and just waiting for some kind of signal to tell it to contract. And then there are the smart doers over here. These are the electrical cells that will actually tell the muscle cells, go contract now, right? So this is the other 1% of cells. So this is 99% of cells, contractile, but the little 1% are the electrical cells. And these are the doers. These are the ones that generate the electrical signal. And so the first set that I need to talk to you about are called pacemaker cells. Pacemaker cells are what we call self-depolarizing cells. Do you remember what depolarization is, right? When the membrane goes up, right? That's a depolarization. These cells in the heart are self-depolarizing. Usually we said like a muscle receives a neuron signal and then it tells it to depolarize. These are self-depolarizing on their own. They have a rhythm, right? A pace that they will depolarize at, right? So here you see an action potential, and then it goes back down. And then instead of staying at this low below threshold, on its own, it's a bit leaky, and it'll self-depolarize. This is called the pacemaker potential. It goes up slowly, it reaches threshold, and it goes on its own. And then it goes, and another bump. It's interesting. They self-depolarize, right? The heart actually has these cells that self-generate electricity, right? They will generate these little waves over and over. And that happens in two regions. In the sinoatrial node. So over here, the sinoatrial node is right here. A node is a little bump, right? Do you see this little bump of electrical cells here? Sinoatrial node, or we call it the SA node, okay? Because it's in the atrium, right? It's the atrial node, sinoatrial node. And then over here we have the AV node, not the AV valve. The AV node is a grouping of electrical cells in this little bump here, a node. This is called the atrioventricular node, AV node, because it's between the atrium and the ventricle, right? So you have these two nodes, electrical nodes, full of these pacemaker cells. So these nodes generate their own electricity, right? And then they send it down through conducting cardiac cells. These are not necessarily generating electricity, but they're carrying the electricity down, right, from the source that it's made. So the electricity is made at these places, and then the conducting cardiac cells, they send these, there's the internodal pathway, the AV bundle, the bundle branches, and Purkinje fibers. So let me go through it basically one by one. Over here at these pacemaker cells, they actually generate an electrical signal on their own. That goes down this internodal pathway, which is part of the conducting cardiac cells here. Internodal pathway because it's between the two nodes, right? Here's the first node, here's the second node. So the electrical signal is made in the SA node. It goes down through the internodal pathway. That'll stimulate over here an electrical signal to be made in the AV node, right, which will go down the AV bundle. It's a bundle. And then though that bundle splits into two bundle branches, right? Do you see how there's two bundle branches, which then each veer into what we call the Purkinje fibers, one on this side and one on this side, right? So basically, right, the pacemaker cells, these are the ones that are making the electrical potentials, and then they just send it down the wires, right, all the way down. I really like this diagram here because you see that it starts over here in the SA node, it goes down the internode pathway to the AV node, which then goes down this AV bundle and then into the bundle branches here, and then up the Purkinje. And all of this to say that these electrical cells are what allow for the heart cardiac muscle cells to contract. Remember, I told you if these were just sitting on the table, they wouldn't do anything. If you sent a neuron to them, then they would contract, right? Remember, even our skeletal muscle, we need our brain to send a signal down to contract it. Same thing here. These electrical cells in yellow, they need to fire to stimulate the walls of the heart to contract. So I really like this picture because I want you to imagine, you see all this yellow? That's the electrical cells, right? Yes, the pacemakers that actually generate it and the conducting, but who cares about that? These are the electrical cells. They do not contract. They just send electricity. But the electricity stimulates all of this. Do you see this light gray all in between the yellow, right? There's this light gray even up here in the atria, right? You see this light gray. All of that would be this stuff, the contractile cells. So as the electrical signal goes through these wires, it is telling the gray walls, right, which are the contractile cardiac muscle, to contract. So I really like this picture. If you slowed it down, you would see that as this yellow electrical signal passes, it will make this gray wall contract, right? So the electricity passes and then it contracts because the electricity tells the muscle cells to contract, right? And then down here, do you see that as the electricity passes down, right? Soon after, the ventricles contract, right? You see, and then the ventricles contract. This is why the heart goes like this. Boom, 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 boom. Because there's a bit of a delay, right? First, the signal starts here, and then it goes down, and then the ventricles contract, okay? So this is very cool. Now, how is the heart stimulated to contract? There are two kind of systems that stimulate the contraction. The heart's electrical wiring is referred to as the intrinsic system. So the pacemaker cells in the nodes are self-excitable or self-depolarizing. Basically, they are autorhythmic. They create a rhythm on their own, and they trigger an electrical signal, right? Myocardial cells will depolarize rhythmically due to this electrical signal next to it, and they will have these coordinated contractions, right? So basically, what I'm telling you is the intrinsic system is the fact that the heart has this SA node that fires down the internodal pathway to the AV node, and then it fires down over here these bundle branches down up the Purkinje fibers, right? And that is the intrinsic system. If you took, and this is why I put this little picture here, if you took a heart out of someone, right, and put it, this is not quite accurate, if you put it in a jar full of ions, sodium, potassium, and all of the good ions, give it some glucose because it needs to be fed, the heart would contract on its own. It would go off on its own and contract, okay? It's crazy, but it's true. This is the intrinsic system. The heart has its own mini nervous system. It is going and starting an electrical signal and sending it down on its own. Remember, I told you the nodes are self-depolarizing? So the heart has an intrinsic conduction system. It conducts electricity and contracts on its own. It is one of the most independent organs from the brain. The brain controls everything and is kind of snobby and controls everything, but the heart is one of the ones that actually has a more dependent, its own independent system for electrical conduction. So the heart can pump without a nervous system as long as it gets the proper ions to do so, and the glucose, right? Now, there's another system though, the autonomic nervous system. Remember the ANS? That's part of the brain. Remember the sympathetic or parasympathetic? This acts as what we call the extrinsic system, and it communicates to the pacemaker cells. So the extrinsic system comes from the brainstem, and it goes over here to the heart pacemaker cells, right? And it will, in the case of the sympathetic system over here, the sympathetic cardiac nerves, they will increase the heart rate and force of contraction. Think of when you're stressed, right? Sympathetic stress. You will have your heart rate increase, right? So the sympathetic system can increase our heart rate and our stroke volume, meaning the contraction. The parasympathetic nervous system, like here's the vagus nerve, that decreases your heart rate. It slows down. Think if you're meditating and cool as a cucumber, right? And that will really calm down your parasympathetic firing. So here's the deal. I want you to think of the heart as a radio. The heart is a radio, and the radio is always playing, right? It is always on. It is always contracting on its own. It doesn't need anybody else. But as a driver, I could turn up the volume and make it beat faster. Or as a driver, I could turn down the volume and make it beat slower, right? So that's the idea here. The intrinsic system, the radio is always going, but I can modulate the volume up or down with the nervous system. So I could go faster or I could go slower, right? So the intrinsic system is like the radio, and the extrinsic system is just tweaking the volume, turning up the heart rate or down, okay? Now, what are the pacemaker cells of the heart, right? We've already discussed this. The pacemaker cells, the ones that auto-generate and make the electrical signal, is the SA node, which occurs in the roof of the right atrium. They will self-depolarize due to their leaky membranes, and this causes action potentials to fire rhythmically on their own. Over here, another group of pacemaker cells occurs in the floor of the right atrium. This is the atrioventricular node, right? Remember, node is a grouping of those electrical cells. So I want us to now just piece everything together and consider the pathway taken by the signal. So what are the electrical cells of the heart, right? First, we go through the SA node. This is located in the wall of the right atrium. This is the pacemaker of the heart, right? Then we go down through, you don't see them very well, there are these little wires that pass the electrical signals to the atrial contracting cells, right? So here, this is just generating it. Then we have these wires that go through the atrium. This is called the internodal pathway. It's wires between the two nodes, but think of what it's going to do. They pass along the walls over here of the atria, right? And they tell the atrial contracting cells to contract. So one thing I'll keep mentioning is that electrical signals lead to muscle contraction. I won't shut up about it. When you send a signal down your brain, I want to contract my biceps, that goes down the electricity to the muscle, and then you tell your muscle to contract, right? I know it seems very quick when we do it in our day to day, but that is the order of events. Electricity tells our muscles to contract. So here, this electricity tells the atria to contract. Then it goes to the atrioventricular node. This is located in the junction between the atria and the ventricles. And it will go down the atrioventricular bundle, this part here. This is called the bundle of His also. That's another name for it. And then it will split into two, the bundle branches, one branch here and one branch over here, right? Down the septum. And then once we veer away from the septum, we call these Purkinje fibers, right? These are my Purkinje fibers, one on the left side and one on the right side. So that's the order that the electrical signals go down. So what are the sequence of electrical events that occur? So results of an ECG, an electrocardiogram, they can show us the depolarization and repolarization events experienced by the cells of the atria and the ventricles. So I want to make something very clear again. Electrical events are fast, physical events are slower, and they also, the physical events, they occur due to the electrical events, so it does take, there's a delay, right? I want you to just look at this animation. You see that go down, right? I want you to notice the electrical signal passing along all of these atrial cells. They will stimulate all of these atria muscle cells, right? But you see there's a delay. Do you see bzz, and then the atria contracts. Same thing for the ventricle. Here, right? You see that the ventricle, the signal goes down and then it contracts, right? That's what I mean by the electrical events. All these yellow things happening are slower than the gray muscle contracting, right? So we're gonna go through the steps over here. The SA node activity and atrial activation begin over here, time 0. Step 2, the stimulus spreads across the atrial surfaces, right? It moves across all of here, right? And reaches the AV node. It'll reach this AV node over here, right? So it'll get to the AV node. This is 50 milliseconds later, okay? So right now at 50 milliseconds, we've reached the AV node. This little arrow will go all the way to here, right? So this will go through the internodal pathway, right? Like the internodal wires to the AV node. I want you to notice the purple is trying to mimic contraction, okay? Do you see that only, like, even when we get to the AV node, right? Like that signal's there. The purple has still not like contracted all over here, right? Even though the signal is at the AV node right now. So that wave comes after. Like over here, you see that the atria are contracting. Do you see how it's more dysmorphed here? That's after all of this happens. So there is a delay between electrical and physical events, right? One thing that's important, this does not pertain to this statement here. This is a separate delay. There is a 100 millisecond delay at the AV node. Over here at the AV node, there's a bit of a pause, okay? As you see here, you see that this sends down, and then it sends down the rest, right? Down, and then down. So there's a bit of a pause at this AV node due to its physiology. We don't need to know why, but it actually makes a lot of sense. And as I said here, the atrial contraction begins once this signal gets all the way here, and then the atria contract, right? Because there's that delay. But the pause that happens, right? The pause that happens at the AV node is important because the atria contract, and then the ventricle contracts, right? So the signal goes through and the atria contracts. There's a pause here before it goes down to the ventricle. So we allow the atria to contract and then the ventricle can contract, right? So there's this delay over here. And then, so 150 milliseconds. Then the impulse travels along the interventricular septum down over here, right? Within the AV bundle and the bundle branches to the Purkinje fibers, right? The Purkinje is on the sides here. And blah, 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 to the papilla. So that will go over here. And the impulse is distributed by the Purkinje fibers and relayed throughout the ventricular myocardium. So all of this stuff, right? The actual impulse goes up the sides, right? And this is where, when these contract, that's where we get a squeeze, right? So the atria contraction is completed and the ventricle contracts. That's when you're going to get the ventricles contract. So do you see how over here the atria contracted, and then later on here we're going to have the ventricles that will eventually contract. So there really is this like double pump, right? I really like this slide. You don't need to understand the graph that much as long as you understand the concept that electrical events occur before physical contraction events. Because here you see that the electrical signals that are sent along the conduction system, they trigger the physical contraction events in the cardiomyocytes, in the cardiac muscle cells, right? I love this picture because do you see in yellow? That's all this electrical signal. Bzz, bzz. In red, do you see all this red? I want you to think of that as the cardiac muscle. The cardiac muscle is what will actually be contracting. The yellow does not contract. It just sends a signal which talks to the muscle around it and gives an electrical stimulation to the muscle around it. They first, the cardiac muscle, right? So the electrical signal sent along the conduction system triggers the contraction of the cardiomyocytes. The cardiac myocytes, right? They first depolarize, right? Remember depolarization and repolarization? Here, they first depolarize up, and that's because sodium rushes in the cytoplasm to trigger a muscle contraction, right? I want us to remember this. Remember that when we depolarize, a bunch of sodium rushes into the cell, right? So this is the sodium rushing into the atrial muscle cell. And then they repolarize, potassium rushes out of the cytoplasm to trigger muscle relaxation. Part 2, all of this potassium here, right, will rush out and it'll stop the stimulation. It'll go down and that causes the muscle to relax, right? So sodium rushing in triggers the contraction, right? And then it relaxes when the potassium goes out. So why do I like this picture? Because it really shows the electrical events and their timing. This should have an axis here that says time, right? And don't worry about this axis, the y-axis here. But over time, right, you see that the SA node here, it has this potential, right? It has this electricity that gets sent through, and that will go down the internodal pathway to the AV bundle. And even over here, you see that eventually the AV node over here will go and it will be stimulated, right, at this time over here. But you really see that the first event that happens is over here. The SA node fires. That's the first event. And what does that cause? That causes all this muscle here in the atrium soon after, right? This muscle will contract. Sorry, it'll depolarize and it'll contract, right? Then the AV bundle, it depolarizes, right? And it sends the signal down over here. And soon after, over here, right, the ventricles will contract, right? A little delay. All the ventricles here on the side, they will contract and they will depolarize and repolarize, right? So I want you to note that there's a delay, right? Electrical events are completed in 225 milliseconds. All the electricity goes through. But the physical events, the actual beating of the heart, that's completed in 800 milliseconds. So there's this lag because it takes, the electricity needs to send the signal to the muscle. The muscle, remember all the steps in from the axon terminal through the muscle, the channels will open, a depolarization happens, the sarcoplasmic reticulum releases calcium. Calcium binds onto troponin. Troponin will move trypomyosin. Trypomyosin allows actin and myosin to bind together. Then there's contraction, there's pulling of the fibers. All of this takes so much time. Just telling you about it took like a bunch of time. So imagine it happening, right? So that's why there's a delay between electrical events and physical events, right? There's a little lag, okay? So I want you to pause the recording and try to attempt this problem over here. Let's go through the steps. So the pacemaker cells, right, they initiate an action potential. What happens next, right? So let's start at the SA node. Then we're going to go and action potentials will go down, right, and distribute and pass through the atria, right? So that would be the next one, E. Then we actually reach the AV node, that's D over here. And remember, in the AV node, there's this delay. There's this 100 millisecond approximately delay. That's the most important because we want our atria to contract, delay, and then the ventricles contract, right? So we need this little delay here. That's due to the AV node. And then once we exit the AV node, we're going to go down the AV bundle. So that's going to be G over here. And then the action potentials will move and split into the right and the left bundle branches down the interventricular septum. So that's A. And then it'll go up the Purkinje fibers and distribute the action potential along the sides. So that's B. And then lastly, the action potential is passed through the contractile cells of the ventricles. All of those contractile cells will get the action potential. And lastly, the ventricles will contract, okay? So now we'll talk about the ECG, the electrocardiogram. Some people also call it the EKG, okay? That's the same thing. So what is the electrocardiogram? It's when you place electrodes on the surface of a patient, right? And you can monitor the electrical activity of the heart. So actually, you can graph out based on what this is detecting. This is detecting these changes in millivolts inside your body, the heart, right? Over time, right? So it's measuring the changes in this electrical activity over time. So it's measuring, basically it's measuring what is the millivolts coming from. If you don't move and you don't move your muscles too much, right, then we can measure this. This is all the action potentials generated by the nodal and contractile cells, right? So it's all of those nodes and all the contractile cells that we're measuring their electrical activity, all the ions moving in and out, the depolarizations and repolarizations, all of these things that are going on electrically we can measure, right? So the ECG results can assess the performance of the electrical components of the heart, okay? This graphic representation, however, I need to just give you a warning. If this doesn't make sense too much, don't worry. But for those of you who are really delving in, when you see these ups and downs, right, you'll notice that these here, they look like action potentials a little bit, right? Remember we saw action potentials look like this, right? They kind of look like that, not quite. I'm just going to warn you to not compare this too much to action potentials. A lot of people like to, we taught you this, that when you see this uptick, right, that's a depolarization, meaning sodium rushing in. And then this is a repolarization, that's like potassium that's rushing out. I would not think of that, because that was for a neuron, one single neuron, measuring the inside versus the outside. We're not doing that, okay? What we are doing is we are looking at the 3D representation of many electrical cells and how the voltage is changing of all of those cells. So we're not measuring one specific cell's depolarization and repolarization. We're looking at a three-dimensional idea of all of these cells. So it's not as easy as up and down, depolarize, repolarize. What I want you to note is if you see a bump or a change or a spike, right? That just means a change in the electrical activity, not necessarily depolarize, repolarize. Well, we will tell you what it is, but just the physics of it and the direction of the bumps don't really matter for us, because we're not learning about the physics of why the bump is in one direction versus the other. I just want you to know, if you see a bump, there's some kind of an electrical change that we're measuring in the heart. So let's look at the waves of the ECG. Okay, so in the electrocardiogram, we measure the electrical signal and not the contraction itself. An ECG is measuring volts, voltage, not pressure, contraction. The voltages do eventually, with a little lag, lead to contraction, right? But it's not direct. You cannot say that you're measuring the contraction. The ECG will tell you if the electrical system is working well. And usually if the electrical system works well, then the contracting system works well, right? So I do understand this correlation, but we are not measuring squeezing, right? When we see a bump like this, that does not mean squeeze, right? This means electricity passing somewhere in this system, right? So that's why I was stressing this electricity stuff happens before the contraction stuff. That's why I was emphasizing that so much. This is the electrical stuff we're looking at. So, for example, the P wave, right, is caused by the depolarization, electrical stimulation of the atrial cells. So all of these cells here, right, in the atrium, they will be depolarized, and that will cause this P wave, right? This electrical activity of these through the internodal pathway will cause eventually the atria to contract, right? Eventually the muscle cells here will contract thanks to the depolarization of the atrial electrical cells, right? But that only happens after. So actually this causes the atria to contract at the top of the P wave. This over here, this is where the atria starts to contract. Atria starts to contract, right? Then you see that this is the electrical conduction, and then there's this delay. Do you see this delay over here? That is at the end of the P wave, there is a pause, right? Because we know that over here at the AV node, there's a little break, right? Since the AV node depolarizes more slowly, right? Why does this pause happen? Because it allows for the time for the ventricles, as the atria squeeze, the ventricles collect blood, right? During the atrial contraction before the ventricles contract. Imagine if the atria was squeezing out the blood and at the same time the ventricle was pushing, they would fight with each other, right? That would be good. So it's good that we take turns. Fill the ventricles, then squeeze. Fill the ventricles, then squeeze, right? It's as if you were trying to water the plant while putting the water in the bucket at the same time, right? You need to first fill up your bucket, then you water your plant, okay? So that's the delay. Then we have this QRS wave, which is caused by depolarization of the ventricle cells. So do you see all of these electrical cells here, right, in the ventricles? That's going to be the QRS wave. All of these bzz, 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 up to the Purkinje fibers, right? So that's going to be caused by the depolarization of the ventricle cells. So all of these cells will depolarize, right? So let me actually, I'll go and remove this for now. This over here, this bump, was due to atrial depolarization. Atrial depolarization. Depol. Okay. That will cause, oops, I'm on the wrong thing. This wave here, right, will cause atrial depolarization. Okay. And this whole thing is due to ventricles depolarizing, right? We see this bump because there's actually, what is depolarization? It's sodium rushing in, right? So we see sodium rushing into the cells over here, and then we see sodium rushing into the cells over here, right? This will cause, atrial depolarization will cause contraction of atrial cardiac cells. Ventricular depolarization will cause the contraction of ventricular cells, right? It will cause it, but it doesn't do so right away, right? There's a bit of a lag. So I just want to write down the electrical events. Ventricular depolarization, that's an electrical event. Now, I'm going to write something a bit weird. Over here, at the same time, you have repolarization of the atria. What goes up must come down. What contracts must relax, right? And the way that a muscle relaxes, right, is that first there's this, first we have a depolarization, which allows contraction, and then things repolarize, which relax, right? So the atria depolarize here, they contract, and then at the same time, in this area, the atria at the same time will go and repolarize. All right, so that's going to happen at the same time as the ventricles depolarize. Eventually, this means, like, what does this lead to? That means that the ventricles depolarize and they'll start contracting. So that means that the ventricles will start contracting as the atria starts to relax, right? Remember I told you that the atria contracts and then the ventricles contract. Atria, ventricles. So at the same time as the ventricles will depolarize, they get all that electrical signal, right, that's moving around to contract. The atria repolarize and start relaxing at the same time. This is why this wave is so weird. Do you see this nice P wave? Very simple, up and down, right? Because one thing is happening, the atria are depolarizing. This is a weird, these two weird events, because I'm not a physics expert, but how I justify it in my head might not be super accurate, but there's like two bumps ongoing that are countering each other, right? There's like this bump over here where the atria are repolarizing and creating this current that you're measuring with the voltage. And there's this bump over here, which is much bigger, right? That looks like this, and it is going upwards, right? So it makes when two waves come together, they mesh and they make this weird wave that you have. Okay, so anyway, that's my way of justifying it in my head, that this wave looks a little weirder. But that's the idea here, is that there's actually two events that are hidden in this wave. So let's go read what I put over here. The QRS is caused by depolarization of the ventricles, but also repolarization of the atria. And what will that cause? That will cause the atrial diastole, meaning atria relaxing. For diastole, you could write just relaxing, right? Which is caused by the repolarization of atrial cardiomyocytes. So because the cardiac cells repolarize, okay, these atrial cardiomyocytes, that can cause atrial diastole, relaxation. It also will cause ventricular systole, ventricular contraction, right? Because this is caused by the R-peak depolarization of the ventricular cardiomyocytes. So because over here we are in ventricular depolarization, then the ventricle will eventually, systole means to contract. It'll eventually contract, right? Lastly, so I said what goes up must come down. The atria went up and then they went down. They depolarized, then they repolarized. The ventricles went up, they depolarized here. They need to go and repolarize too. This T wave here is showing ventricular. My colors make no sense right now, so just don't worry about the colors. Ventricular repolarization. So over here, right, T wave is caused by ventricular repolarization, which will cause relaxation of the ventricular cardiomyocytes, and they'll relax and go back. So here my atria depolarize, which will cause the atria to contract. Then over here, right, the ventricles depolarize, which will cause the ventricles to contract. And at the same time as the ventricles contract, my atria is repolarizing, which relaxes, right? So contract. And then eventually my T wave, my ventricles repolarize and they relax, right? And then the cycle goes back. Then the atria squeeze, right? And then it goes through. So this is really just this whole heart ECG over here, right? It's just explaining one cycle of the cardiac cells. So boom, 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 boom. It's just giving boom, boom, right? That's all that it's saying. When the atria and the ventricles are contracting, that's the idea that we're given in this one snippet. But remember, this gets repeated. Now, for the intervals of the ECG results, these aren't the actual waves, the bumps. These are actually like a certain time that we are measuring, and they could be very useful clinically. So over here, the PR interval is the time from the beginning of the atrial depolarization, so the beginning of the P wave, to the initiation of ventricular depolarization. Where does the ventricle start depolarizing? At the QRS complex, right? So over here, sometimes it's the Q wave, sometimes it's the R wave they measure. So I know it says PR interval, sometimes it goes to the Q, sometimes to the R. Basically, since the Q wave may be difficult to view in a tracing since it's so small, the measurement is often extended to the R wave, which isn't that far off, and it's more easily visible. So that's why they call it PR interval, but technically they measure up until the Q wave. And why is this measure important clinically? Should there be a delay in passage of the impulse from the SA node to the AV node? Let's say something is off in the circuit from the SA node to the AV node. It would be visible in this PR interval. If it's shorter or longer, then there are maybe some issues in that part of the conduction. The ST segment is the time between the end of ventricular depolarization, so the end of the QRS complex, right? So right here, and up until the beginning of the T wave over here. And this measures the time the ventricle contracts to expel blood out of the ventricle, right? It's technically the time that that ventricle is actually contracting. So, why is this important? Because we want to be squeezing the ventricle as much as possible to contract the blood out, right? And send as much blood out as possible. If this amount of time, this segment is changed shorter, right? Then we won't be squeezing for as long. We won't be expulsing as much blood. That's a problem, right? That's not good because really, like, we want to be maximizing the amount of blood that we send out. So changes in the ST segment actually, if a patient has some signs of a heart attack or different heart conditions, this ST segment could be one of the measures that they look at to see in the ECG. So here's the sequence of events in our ECG. Remember, the ECG measures the electrical signal, not the contraction itself. So this is a good summary of, looking at the electrical event, comparing the physical event, okay? So first, the SA node generates an impulse and the atrial excitation begins, right? So in the P wave over here, right at the beginning of the P wave is when the SA node will fire, right? So the SA node depolarizes and generates an impulse. The internodal pathway depolarizes all over here, right? That internodal pathway. And that will cause the atrial myocardium to depolarize, and that will cause eventually the atria to contract, right? Eventually. Over here, right at the end of the P wave, the AV node depolarizes, right? This is when at the end over here, this AV node will start to depolarize. The impulse is delayed at the AV node, right? There's this, do you see this flat line between the end of the P wave and the beginning of the Q wave, right? So that delay is because the AV node takes its time to actually send its signal. The reason why is because the cells are smaller in diameter and than the conducting cells, but you don't need to know that fact. You don't need to know that specific fact. Just know that there's a delay at the AV node, so the impulse is delayed at the AV node. That's why we get this little flat line for a while, right? And then at the end of the P wave, up until the Q wave, right, the impulse will start to spread rapidly through the AV bundle. So it'll go down through the AV bundle, right? The impulse passes to the apex of the heart. And ventricular excitation begins, right? So it'll go through the bundle branches to the apex of the heart here. And then in the QRS complex, right, the Purkinje fibers depolarize. These fibers over here will start to depolarize. And the ventricular, over here, the ventricle walls, right, the ventricular myocardium will depolarize and will actually fire. So that's why you see this QRS complex over here. So that's your ventricular excitation. I really like this slide because it shows the ECG, the PQRST waves, and it shows in blue the electrical events, right? That pertains to the ECG. That's why it's in blue. So over here, the P wave showing the atrial depolarization. The QRS complex is both the ventricular depolarization and the atrial repolarization, right? And then the T wave over here is our ventricular repolarization that's occurring. And at the same time, over top of that is the physical events that occur. So over here, you see in red, 2 bumps, right? So from the middle of the P wave, you will start to have the atria contracting, and it reaches its maximum peak contraction over here, and then it goes back down and ends about at the R wave. Whereas the ventricles will contract starting at about the R wave over here, and it goes and reaches its maximum contraction over here, and then it starts its contraction starts going down, right? And at the end of the T wave, it will stop. So this could better be seen over here a bit more clearly. You see anything that says systole means contraction. Diastole means relaxation. Okay, so over here you see very nice. The atria and the ventricles, they are, independent. Over here you see that the atria are in relaxed mode in diastole up until the top of the P wave, right? Because the P wave is what's going to allow the electrical signal to pass everywhere, and then right about halfway through the P wave is when the atria start to contract, and they stop contracting here, as I said, at the top of the R wave, and then they go into relaxation mode, right? Diastole, atrial diastole. On the flip side, right, remember I said that the atria contract and the ventricles are relaxed. When the ventricles contract, the atria are relaxed. And you see this very well because when the atria contract, the ventricles are in diastole, in relaxation, right? So over here, right, as the atria are in systole, squeezing, right, these ventricles are in relaxation mode so that they can pick up all that blood. So this is ventricular diastole up until the QRS complex, because the QRS complex is what will depolarize the ventricles, right? So right at the top of the Q wave, we say that we start ventricular systole, contraction, and it goes all the way until the end of the T wave, right? Because here we have the repolarization. And then we're in ventricular diastole, right? So it goes boom, 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 boom. It has this kind of offset beat. Now, this is the section where we piece everything together, okay? The valves, the chambers, the ECG. So I want us to take a look over here at this slide. What causes the valves to open, right? The answer is pressure. Blood pressure between two chambers is what causes the valves to open. So I want us to look at this graph over here. Okay, this examines the flow of blood as pressure builds up. All right, so on this axis here, you have pressure, and on this axis, it should say time. So this is pressure over time. Now, what I need you to do is to compare two lines at a time. The blue line is the pressure in the left atrium. The red line is the pressure in the ventricle. And the black line is the pressure in the aorta. Okay? So, when do valves open? Let's take a look. First of all, we have two types of valves, right? We have AV valves and we have semilunar valves. So over here, let's say I have my heart, right? I'll draw the two chambers, one, and then over here you have the atria, right? So here's our septum. Let me draw first. I'm just going to draw it on one side because the other side is exactly the same. The AV valve, right, is, I'm going to actually draw properly. The left atrium is in blue. Do you see this blue line here? That's the atrium, so I'm going to go and draw this in blue. The left ventricle is in red. So over here, right, in red, 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 red. This was in blue over here, and then this is, this is my, we're going to draw it like this. There we go. Blue over here. All right, so here I have my left ventricle, left atrium, and over here my aorta, okay? So when is it? This is showing the fluctuations of different pressures. I want you to look at it one line at a time. Over here you see the line, right? And do you see this bump over here, right? This bump is due to the atrium that is squeezing and increasing the, I want you to imagine being a little, for the blue line, I want you to imagine being a little red blood cell over here in the atrium and getting squeezed. And you see that this bump, this line goes up like this, right? For the red line, I want you to imagine being a little red blood cell over here in the ventricle, right? Do you see over here the line? It goes and it shoots up. Now, I want you to know that the bump for the atrium is smaller than the bump here for the ventricle because the atrium doesn't squeeze very much and it's a thin wall, whereas the pressure in the ventricle is super high. The wall is thicker and so it squeezes even tighter, right? So that's why the red line is, the ventricle is bigger than the atrium, right? But now that we have looked at that, I want you to remind yourselves where the AV valve is, right? The AV valve is over here. Actually, I'll draw it on this side. It is between the atrium and the ventricle, right? It's over here between the atrium and the ventricle. So, when does the AV, remember I said that valves close just because of pressure differences, right? So over here, do you see that the blue line is above the red line? That's because the atria is in systole, right? It's contracted and the pressure inside here, there's a lot of blood pooling into it. There's more pressure in the atria and the ventricles are just open and relaxed. You would much rather want to be in the ventricles that are relaxed and just dribbling blood in than in this atria over here squeezing. So that's why the pressure, the blue line, is above the red line at this point, because we're still in atrial systole here. Then all of a sudden, right, at this very moment, you see the red line go and shoot up compared to the blue line. So the red line, the ventricle pressure in millimeters mercury, right? The ventricle's pressure goes bigger than the blue atrial pressure. Why would that be? Because probably at this point here, the ventricles will now start to contract and the atria are relaxing, right? Do you see how the atria, the line starts going down over here? So the atria is relaxing and the ventricle starts squeezing. And remember, the ventricle has a thick muscle wall. So when it squeezes, it really creates a huge pressure. And so that spikes up, right? You see the pressure in the ventricle spike up. Now, we don't really care about that for now. What we care about is that the AV valve, all that it cares about is whose pressure is bigger than who, right? So the AV valve will be closed when the pressure is greater in the ventricles compared to the atria. So over here, when the red ventricles start to squeeze and they beat the pressure of the atria that are then going to relax, right? When this pressure is bigger than this one, that's when the AV valves will close, right? They're going to close like this. So up until here, right, all of this region, right, the AV valve is open because the atrial pressure, this pressure here is bigger than the ventricle pressure because the ventricle is just relaxed. And then at this very point is when there's a transition and the ventricles squeeze and that ventricular pressure increases, but the atrial pressure goes down, right? So this is the point where we say the AV valve is closed, because when the pressure is greater in the ventricles compared to the atria, then we have the AV valves close, right? The parachutes, the blood's going to push against these little parachutes and close the valve. Now, the semilunar valves, right, they open when pressure is greater in the ventricles compared to the aorta. Remember, I always told you, you just need to compare the two regions, right? So over here for this one, we are comparing the aorta, right? And remember, the semilunar valves are like this, closed, and they could open up like this, right? They could open up this way, right? So they could, like, I'll put a little arrow, they could open up, they are like this, and they could open up upwards, right? So what's going to happen, we need to compare the pressure. Look at the aortic pressure, right? This aorta pressure is humongous. It's a huge pressure over here in the aorta pushing against. Our blood pressure in our vessels is huge, right? It's over here. And we need to ask ourselves for the semilunar valve, when is it that this ventricle pressure is going to be high enough to burst open those valves, right? Well, the semilunar valves will open when the pressure in the ventricle is greater than the pressure in the aorta or the pulmonary trunk if it's on the other side, right? So over here on our graph, do you see that the left ventricle, think of a water balloon, and you slowly squeezing it, right? The pressure is going to build up, right? Think of there being a straw in the water balloon. If you do a light squeeze, the water might not come out, right? Maybe if you do a medium squeeze, a little bit of water comes out. And if you do a large squeeze, then a lot of water will come out. That's the same concept here. We build up and we squeeze our pressure till it bursts and beats the pressure of the aorta, which is very high, right? It bursts open the doors. So the doors then open up like this and blood can now flow in this way. So this is the point here. Do you see where the red line becomes over here? The red line, let's pick a point, a time point. This time point here, the red line is over here, but the black line is over here, right? But at this specific time point here, right, this is when the black line will end up being below the red line. Do you see how here the black line is below the red line? At this time point, the black line is below the red line, and here it should be kind of like this. Unfortunately, it got shifted a bit. So the red line should be above the black line. That's when the semilunar valves are going to be open, because the pressure in the ventricle beat the pressure in the aorta and bursted the doors of the semilunar valve open. Now, the AV valves, actually, we're going to start over here, the semilunar valves. So remember our semilunar valves. I'm just going to go and draw again the ventricle here. We have the atria over here, right? And then we have, lastly, we have this. We have the vessel, the aorta over here, right? Here's our semilunar valve, let's say. Oops. Let's just go and do that again. One. So we'll draw our semilunar valve. We'll draw it open for now, because right now we're in the open phase. So here's our aorta, and here's our semilunar valve that is open, right? It's open up like this, and its blood is allowed to flow through, right? Well, the semilunar valves, they will close when the pressure is greater in the aorta, right? When this pressure beats the pressure from the ventricles. So do you see how this red line, it goes up, and then it descends, right? Why does it go down? Because the ventricles, they squeeze, and then they get a signal to relax, right? So that's why you see this curve go back down. So when it is at this point and the red line becomes lower than the black line, do you see how here the red line is below the black line? Here too, right? Up until this point it wasn't, but now the red line is below the black line. So this pressure does not beat the aortic pressure. So now this pressure wins and it will close the valve. So let me show you what the valve looks like when it's closed. I'm going to erase these two. Now we're going to close the valve, right? So it's going to look like this. We're going to close the valve like this. So it was open like this, and then all of a sudden, clock, the two valves will close because this aortic pressure beat the pressure down below in the ventricles, because the ventricles are now relaxing, right? And so relax, relax, relax. So the semilunar valves close when the pressure is greater in the aorta compared to the ventricles. Now we're going to look at this one here. So the ventricles keep relaxing, right? And the AV valve, let's go draw our AV valve again. Here's our AV valve. Our AV valve, right now it's closed. Why is it closed? It's closed because at these points over here, right, we said that the ventricular pressure beat the atrial pressure, right? But now the ventricular pressure is going down, down, down. And at this very point, the ventricle pressure, do you see here? It's the red line, the ventricle is below the blue line. So let me go and show this on our little diagram. The ventricle pressure here is going to be lower compared to the atrial pressure. So the atrial pressure wins at this very point, and it bursts open these doors. So these doors will open up like this, right? So now I'm going to go and erase these over here. I burst open my doors at this point, and now you see that the red line, the ventricle, is lower than the blue line because there's the ventricle is relaxing and now the atrial are contracting. Okay? So one thing to note, I've been using these terms a little bit without explaining, so I want to explain these now. I want to talk about contraction and relaxation phases of the chambers. We talk about this contraction relaxation cycle of the atria and the ventricles, right? I said that they contract and relax, contract, relax. Systole is the term that we use for contraction events of the atrial and ventricular cells. These are needed to force blood out of the chambers. When the atria are in systole, they force blood into the ventricles. When the ventricles are in systole, they force blood out through the vessels, like the aorta or the pulmonary trunk, the arteries, right? Diastole is the term that we use for relaxation. These are events of the atrial and ventricular cells that relax, and these are needed to allow the chambers to refill with blood. So for example, when the atria are in diastole and they relax, that's to allow blood to come back into the atria and fill up the atria again with blood, right? When the ventricles relax, that's to allow the ventricles to fill back up with blood. So this is our systole, diastole. If I go back to our ECG, you'll see these terms, atrial systole, it's the same thing as atrial contraction, right? Atrial diastole is the same thing as atrial relaxation. I want you to understand that the atria and the ventricles, they relax and contract at their own rate, right? Actually, I keep telling you that when the atria contract, the ventricles are relaxed. And when the ventricles contract, the atria will relax, right? So the systole and diastole are a bit offset. So I want you to pause the video now. This is a very good exercise, actually. I want you to fill this out. So when the pressure is, I want you to even draw it out. Draw out the pressure arrows being bigger or smaller across a certain valve, right? And I want you to tell me that this is saying the aorta pressure is bigger than the left ventricle. What valve are we considering in this case, right? Between these two regions, would the valve be open or closed? And is the ventricle in systole or diastole? Okay, so I want you to pause the video and take a minute to fill this out. Alrighty, so we're gonna do it one at a time. So over here in this case, I'm saying that my aorta pressure is bigger than the left ventricle. Here is my left ventricle, right? Here's my left ventricle. And here is my aorta. Unfortunately, you do not see the semilunar valve, which is over here, right? So the valve affected is actually the semilunar valve. It's between the ventricle and the aorta here. So this is the semilunar valve. But which semilunar valve is it? This is the one that is for the aorta. So it's the aortic semilunar valve. Okay? Is the valve open or closed? Here I'm saying that the pressure in the aorta is bigger. So this pressure here is bigger compared to this pressure in the left ventricle. So when this pressure is bigger, remember that these little moons, right, they do not go inwards. They only go up and down, right? So if the pressure on top is bigger, then they're going to be closed, right? They're not going to be open like this. They're going to be closed because it's pushing downwards onto the cusps. So this is going to be closed, right? Is the ventricle in systole or in diastole? Well, if the left ventricle's pressure is less than the aorta, then it's probably relaxed, right? It's probably in ventricular diastole, in relaxation mode. Ventricle in diastole. And so we are good with that. Now, I want us to do the next one. So the right atrium versus the right ventricle. Remember that this is the right side, this is the left side. So the right atrium is over here. The right ventricle is over here. What valve is at play here? It's this valve over here. This is called the right AV valve. Okay. Valve. Is it open or closed? Here I'm saying the right atrium is bigger than the pressure in the right ventricle. So I'm saying that this pressure is bigger than this one, right? So is the valve open or closed, right? Remember this valve, it is like this, and it closes like this, closes like this. And I'm saying that this pressure is bigger compared to this one, so they would be open, right? So this would be open. Is the ventricles in systole or diastole? If this pressure in the atria is bigger than the ventricular one, then the ventricles are probably on vacation. They're in relaxation mode. So we're in ventricular diastole, right? Okay. Next one. The left atrium and the left ventricle. Here is, I'm actually going to erase, just going to erase this stuff for now. Now we're going to go to the next one, left atrium versus the left ventricle. Here's my left atrium. Here's my left ventricle. And here I'm saying the left atrium is smaller in pressure than the left ventricle. So the pressure in the left atrium, right? This pressure is smaller than the pressure over here. What valve is at play? This valve here. That's between the two regions. That's the left AV valve, right? And is it open or is it closed, this valve? Well, when this pressure is bigger, right? Remember this valve only opens or closes. When this pressure is bigger, right? The blood will push on the valves and push them closed like this, right? The valves do not do this, right? They only go like this. So the pressure will close the valves like this. So that means that the valve is probably closed, right? And if this pressure in the ventricles is bigger than this one, it is not on vacation mode. It's not in diastole. The ventricles are probably in systole. They're contracting and they beat the pressure in the atria, which isn't very hard to beat, right? But they do beat it. So systole. Next, right, over here we have the pulmonary arteries bigger than the pressure in the right ventricle. So the pulmonary arteries, right, we'll look at the pulmonary trunk because the arteries are just over here on each side. That pressure is smaller compared to the right ventricle. Here's my right ventricle. So this is what we're talking about. So the valve at play here is the semilunar, right? Semilunar valve. And which one is it? This is the pulmonary semilunar because that's the one in the pulmonary trunk. So the pulmonary semilunar. And is the valve going to be closed or open? Here, I have a big pressure in the ventricle and a small pressure over here. So remember, these little cusps, these little semilunes, they go up like this. They never go downwards, right? They just go up in this direction. So over here, if this pressure is pushing against it, would open them up and they would open this way, right? So these would be open, right? And are the ventricles in systole or diastole? Here, I mean, the ventricle is probably in systole because here you see the arrows are bigger. There's a bigger push that's beating the pressure over here in the trunk. So the ventricles would not be on vacation. Ventricular systole. Okay. So that's a very good way to think about the valves. I really like this exercise because you really get to think about the valves here. Now we're just gonna piece everything together one by one. And we're gonna look at, we call this the cardiac cycle. It's basically piecing everything together and understanding what's going on. So we're going to use our handy dandy graph as usual, but we're going to piece it to what's going on in the heart. So first, let's talk about the mechanical events of contraction during atrial systole, right? During atrial systole, we're going to start with the atrial contraction beginning, okay? So the right and the left AV valves are open, right? When the atria are in systole, do you see this green arrow squeezing on the atria? The right and left AV valves are open. These valves are open and you see blood flowing down, right? The majority of the ventricles fills passively. So 70% of the blood that's going to be in the ventricle fills passively. It falls down because of gravity. And then atrial contraction, that squeeze of the lemon, you're squeezing the last drops of lemon juice out of the lemon. That comes from the contraction of the atria, which is required to fill the last 30% of the ventricles. Whatever blood is in there, 30% left, right? So that's atrial contraction beginning. Then atrial systole ends, right? And this is, so you see that you're going to stop squeezing the atria, right? And the ventricles now contain what we call the maximum blood volume, right? Because we're done squeezing the last drops of lemon. This is called the end diastolic volume. The ventricles were in diastole. They were in relaxation mode and they were just piling all this blood, and the blood was filling up, right? Well, at the end of ventricular diastole, end diastolic, right? There's a certain volume in my ventricle, right? I like to think it like a bit like a cup of wine, right? The ventricles, you start filling up the cup of wine, right? You slowly fill it up, and then you get to this point over here, right? You get to a point where this over here, right? Oops. This amount that you put in is the end diastolic volume, that you filled up your ventricle up to here. End diastolic volume depends on the length of ventricular diastole, right? How long is it that the ventricle is relaxing? If for some reason the ventricle is prematurely contracting, then you won't get as high of an end diastolic volume. And interestingly, it also depends on the venous return. How much did blood properly get back to the atria to then flow to the ventricles, right? If not, then you might not get as much blood entering your ventricles. So end diastolic volume depends on those two factors. At this point, right, the atria is in diastole for the remainder of the cycle, right? Because now that we're done squeezing, that's it. Now the ventricles do their work, and that's the real job. So the atria contracting, you see that over here. You see the blue line, the blood pressure go up. Sorry, the atrial pressure go up, right? So that's the atria contracting. And then in this phase, you see the atria relaxing, right? In this second phase here, you see the atria relaxing. So number one, contraction begins at 1. 2, atria eject blood into the ventricles for 2. And then 3, atrial systole ends over here, right? And we're going to see that there's going to be the ventricles that'll start to contract. So now we saw mechanical events of atrial systole. Now we're entering ventricular systole and atrial diastole. So the atria experience diastole, relaxation, at the beginning of ventricular contraction. So my atria squeezed, and as I'm relaxing, my ventricles will start to contract. So the ventricles contract and the pressure rises in the ventricles. All the valves are closed, right? And the ventricles will cease to fill, right? So at this very point, right, remember the AV valve closes. So during all of this period, the AV valve is closed, and up until here, the aortic valve was closed, and now it will open after this. But during this time point, right, over here, from here to here, actually all the valves are closed and the ventricles cease to fill. There's no blood coming down from the atria because the AV valves are closed, right? Do you see this valve closed? And there's no blood exiting the arteries because the semilunar valves are closed. So we call this isovolumetric contraction because the heart starts to contract and it is building up the pressure in the ventricles. The pressure is building, right? It's kind of like a water balloon with no straw coming out of it. You're going to squeeze the balloon and the pressure starts to mount, right? Or think of it like even with a straw, right? You squeeze a water balloon and you need to build the pressure before water actually exits the straw, right? That's isovolumetric because the volume inside of the ventricles doesn't change. It doesn't exit anywhere, or there's no blood added to increase the pressure. So we call it isovolumetric. Now, part D over here is called ventricular ejection, right? When the ventricles are ejecting out blood. So eventually, right, from the isovolumetric contraction, we have this squeezing. Nothing is coming out yet. Do you see over here? We're squeezing and this valve is closed, and then boom, the semilunar valves for the pulmonary one and the aortic one, they burst open because the ventricles have squeezed so much, right, that the ventricular pressure eventually exceeds the arterial pressure. By arterial, I mean in the arteries, so in the pulmonary trunk or in the aorta, right? And it opens the semilunar valves and allows blood to leave the ventricles, right? So the amount of blood that's ejected is called the stroke volume. I want you to imagine collecting, here's the aorta, right? I want you to imagine collecting at the bottom of the aorta a little glass of wine and collecting all of that blood that's ejected out, right? That would be your stroke volume, okay? How much is it that each ventricle is pushing out, right? Because eventually the ventricle will stop contracting, and that will be, all of that stuff that exited will be the stroke volume. So we had two measures. So we've seen end diastolic volume. That's how much is it that we fill up our ventricle with. Stroke volume is how much, when I squeeze the ventricle, how much is ejected out, right? And that's not going to be 100% of what's in the ventricle, actually. We're going to see this in a minute. So I want to look at these two things. So isometric contraction, this is the part over here that is isometric contraction because You are, I told you in this isovolumetric contraction, the volume is iso. It is the same. Iso, like an isosceles triangle, means two sides are the same, right? So same, it means the same. So isovolumetric means the same volume is kept. So as the ventricles squeeze, no blood is exiting, the same volume stays in the ventricles. That happens here because the AV valve is closed over here, right? And up until over here, right, is when the aortic valves open. So during all of this time, both the AV valves and the semilunar valves are closed. So we build up that pressure. Then the ventricular ejection occurs as you see the red line, the ventricle is higher in pressure than the black line, the aorta. So it bursts open the semilunar valves. And this is where the ventricle is contracting, right? All the way up until here. Because you see that the pressure is building, 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 right? Then we have ventricular diastole. What does diastole mean again? It means relaxation. So this event is characterized by an early phase and a later phase occurring when the semilunar valves close and the AV valves open. Okay, so let's take a look at that. The first phase is the ventricular pressure falls, right? So the ventricular pressure, remember the ventricles just finished squeezing as tightly as possible, and now we're starting to loosen up the ventricles. It falls until the aortic pressure is greater than the ventricle, right? When that aortic pressure is bigger, right, over here, when the aortic pressure, here's the aorta, right? When this pressure is bigger than the one in the ventricles, then the semilunar valves will close. So this causes the semilunar valves to close. And so I want us to go look at it over here. Here you see that the maximum pressure of the ventricles is obtained here, and then the ventricles start relaxing. And think of being a little blood vessel in the ventricle. All of a sudden you start feeling this relaxation. You start feeling less pressure go down, 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 and boom, the semilunar valve will close, right? So that's occurring. Going down, right? The ventricles now contain what we call the end systolic volume, because now that the ventricles squeezed, right? Now they're relaxing and they're going to stop ejecting blood. So when those semilunar valves close, whatever blood is left in the ventricles, we call the end systolic volume. Because the ventricles are at the end of systole, they're stopping their contraction. They're at the end of systole. And what volume is left? And interestingly, it's about 40%. Okay? So over here, right? Here is my ventricle, right? And it fills up to, let's say, whatever this is, right? Here's my ventricle. Let's call this 100%, let's say, right? This is my 100% amount that's in my ventricles, right? Then it will eject out the blood, right? It'll squeeze out the blood. So this is my end diastolic volume, right? Because the ventricles were in the end of diastole. And then it ejects out, right? Let's say it goes down to about 40%, right? This would be my end systolic volume, right? This is what's left in my ventricles at the end of systole, 40%, which means that if I put a little wine glass over here, right, and I collected all the blood that was ejected out, right, this amount here, right, in this particular case, 100% minus 40%, I'd have 60% of the blood that got ejected out. That would be my stroke volume. That's how much got ejected out, right? So actually, we have this formula that says that the stroke volume equals the end diastolic volume minus the end systolic volume, ESV, okay? So that's our little formula for this. Now, so that's interesting. Once the valves close, then we didn't squeeze 100% of the blood out. There's about 40% of the blood that still stays there. So that's over here, right? Cluck, closing of the valves, and you can measure then how much is left, and that's your end systolic volume. But then remember, the pressure keeps falling, right? This is just the pressure fell, we released a little bit, and we got to over here, right? The valves closed, but then the ventricle will keep relaxing. It'll keep going down over here. So pressure keeps falling. Ventricular pressure is still higher than the atrial pressure, right? Do you see how over here the red is still bigger than the blue atrial pressure? That's because it's still going down, right? Technically, in this section, all of the valves are still closed. The semilunar valves are now closed, and the AV valves are closed in this region number 7. So all the heart valves are closed. We call this ventricular, the ventricles are relaxing. It's isovolumetric relaxation, because since all our valves are closed, we're not sending blood out to the arteries. We're not collecting blood from the atria. It's the same volume, and we are releasing the pressure in our water balloon, right? Isovolumetric, no blood is added or removed, and we're relaxing now, right? ISO, the same volume is staying in. We're not ejecting or bringing in new blood. But then, right, then we hit this point here. Do you see this point where the two cross and the ventricle's pressure gets lower than the atrial pressure, right? This point right here is when your AV valves will now open up, right? So the atrial pressure, because the atria will start to squeeze again, is going to be bigger than the ventricular pressure, right? So the AV valves will open. And we're passively going to fill in the atria and passively fill in the ventricles, okay? So that's going to be the situation over here, all right? I think that is it for this. If you would like to review this, right, go through this diagram over here, each of the phases one by one, and they write a little bit of text over here to kind of guide you through that. Now, one thing to note is the heart sounds. So the heart sounds, if you have ever heard a heartbeat, right, you'll hear this boom, 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 boom. There's these two sounds, sound 1 and 2, and we call them the lub and the dub. Lub, dub, lub, dub. The first heart sound, the lub, results from the AV valve closing. Sounds don't come from anywhere, right? If you squeeze a water balloon, it doesn't make a sound. It's the valves closing that make sounds. S1 and S2. Valves, they close and they clap, right? They quite literally are clapping for you in your body. They're hitting each other, and that makes the sound, okay? So the first heart sound results from the AV valves closing at the beginning of ventricular systole. So the first heart sound occurs right over here, okay? So do you see when here the blue line is the ventricle? When the ventricle, right, pressure becomes bigger than the yellow atrial pressure, this region right here is where you hear the first heart sound, because that's when that ventricle starts to squeeze, that's when the AV valve will close. That's the first heart sound. That's the lub. So over here, do you see the tricuspid, or the right AV valve and the left AV valve, the tricuspid and mitral? When they close, right, that's when we get sound number 1. Sound number 2 comes when the other valves close, right? They don't happen, we don't have any noise. When we clap, right, the noise comes when the two hit each other. So when the valves close, they hit each other and they make a sound. When they open, they don't make a sound. So closing of the AV valve for sound number 1. And the cuspid valves, right, when they open, they don't make a noise, but when they close back, they make this noise. So over here, do you remember that this is where the semilunar valves, they will open, right? But then right over here is where the semilunar valves close. So you see that the second one is when the semilunar valves close. So the dub sound results from the semilunar valves closing at the beginning of ventricular diastole. So when the ventricle starts to go into diastole, right, and starts mounting down in pressure, when the ventricular pressure gets below the aortic one, that's when you hear the second heart sound. So here you see the aortic and the pulmonary valves, when they close, that's the dub. That's the second heart sound. So I'm going to ask you to pause the video here and try to go through each of these, A, B, C, D, and mark them on the graph that we have here, okay? Pause the video and then play it once you are done. So the first thing to note is to circle the areas of the plot where the atrioventricular valves will be open. These are the AV valves, right? So the AV valves, we know that they're going to be open when the pressure, first of all, if you're not sure, remind yourself where the AV valves are, right? So over here, this is the semilunar, right? Here's my semilunar valve. Here's my AV valve, which is over here, right, between the atria and the ventricle. This AV valve, right, when is it going to be open? It's when the atrial pressure over here is bigger than the ventricular pressure, right? So where is the atrial pressure in purple bigger than the ventricular pressure? Well, the ventricle is red, so the purple is bigger than the red all over here. So I would circle all of this region. That's when the AV valves will be open. And then the red is bigger than the purple down here during all of this section. And then over here, the AV valves open back up again, right? So that's good. I got A. It says put a box around the area where the semilunar valves are open. So the semilunar valves over here, right? When are they going to be open? They will open up when the ventricular pressure is bigger than the aortic pressure, right? Or the artery pressure. So when is it that the red line is bigger than the aorta black line? That's going to happen all over here. You see that the red is higher up than the black up until, I'll put it here, right? Up until this area. So right here is when we have that switch. Place a star where you would hear the first heart sound. The first heart sound is the closing of the AV valve. So the closing of the AV valve occurs when the pressure in the ventricle beats the pressure in the atria. So where is it that the purple line goes above, where is it that the ventricle, the red line, shifts above the purple line, the atria? That would be right over here, right? So that's going to be our star. And lastly, place an X where you would hear the second heart sound. Remember, the second heart sound is the closing of the semilunar. That's going to occur once the pressure in the aorta beats the pressure in the in the ventricle, right? So where is it? The ventricle is in red, right? This is higher than the black line, than the artery, the aorta. And then all of a sudden, over here, right, you see that there's a switch. The ventricle gets lower compared to the black aorta line. So that's the answers to this little problem. We're going to end on just a few last things. How do we regulate the heart? So the first thing I need to tell you about is what cardiac output is. Cardiac output, as we said, was blood flow. It's the blood volume that's pumped by the left ventricle in 1 minute, right? It's the flow of blood. How much is coming out of that heart per minute? And it has to do with the heart rate, the number of cardiac cycles, the number of beats in 1 minute. And it also has to do with the stroke volume. Now we understand what the stroke volume is, right? It's the volume of blood that's ejected out from a ventricle during systole in mils per beat. So when considering cardiac functioning, cardiac output is actually the value that we're most interested in, since this is a measure of blood flow through the peripheral tissues to the entire body. If we have inadequate cardiac output, blood flow, to the capillary beds, this creates inadequate perfusion, which means that the tissues don't get well perfused and there is insufficient exchange of gases, nutrients, and wastes, right? If our cardiac output doesn't keep up, homeostasis cannot be maintained, and that's not good. So a typical healthy adult at rest will have approximate cardiac output, stroke volume, and heart values as follows. Cardiac output per minute. A normal average adult has about 5 liters per minute that is exiting out of the heart. Okay, this should be liters per minute. So think of it, 2, 4, 6. This is these big soda bottles here. These are 2 liters each, right? So it's about this much, maybe a little bit less, right? In 1 minute, right, your cardiac output, what your heart sends out per minute, is this much, right? And where does that come from? That comes from the fact that we have 72 beats approximately per minute. And for every beat, we exit out about 70 mils, okay? So that's where that calculation comes from. And again, heart rate times stroke volume will give you your cardiac output. So 72 times 70 gives us about this much, all right? Because this is the beats per minute, this is the milliliters per beat. So you cancel out the values of beats, right? And you get the mils in 1 minute, right? So that's what cardiac output is. How fast and is it beating? And per beat, how much are we ejecting? You multiply the two and you will understand what the value is of the cardiac output. When is cardiac output modified, right? I said the average cardiac output is about 5 liters per minute. When is cardiac output modified? A healthy individual's cardiac output at rest is about 5 liters, as I said. During exercise, cardiac output will increase to meet the needs of muscles, right? So I said over here, this is the average resting cardiac output. And as we go up, right, the normal range of cardiac output during heavy exercise is somewhere between this 15 and 30, right? Maximum for trained athletes exercising at peak levels is all the way up here, right? This is if you're a really good athlete, then this will be your cardiac output. And actually the opposite during heart failure, right? Cardiac output decreases and cannot meet the needs of the tissues. So this is during heart failure. You see that the cardiac output actually is lower, right? So what variables modify cardiac output? I told you in this formula that we can modify our cardiac output by changing our heart rate and our stroke volume. And that's exactly what happens during exercise, right? You literally feel your heart rate increasing during exercise because it's trying to increase the cardiac output. It's doing this. It used to be at this 5 level, but now it's when you're running, you're going to increase heart rate and therefore increase cardiac output to about this much, right? So we're going to see how that the variables that can modify cardiac output are the heart rate. Here's a normal heartbeat, and we can go faster, right? Beep, 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 beep. Stroke volume is the actual amount of blood that gets ejected out. We can also change that to modify our cardiac output. So we're going to start, so cardiac output, right? Remember, it's the heart rate times the stroke volume. Here's the heart rate and here's the stroke volume. I want us to see how the heart rate can first be modified. The heart rate corresponds to the number of cardiac cycles, heartbeats, the P wave, QRS, T wave, that's one cardiac cycle, right? How many of those occur in 1 minute, all right? You could calculate it yourself by counting your heartbeat, right? The speed of these cardiac cycles will influence the cardiac output, okay? So how fast those are will influence your cardiac output. So think of it, this is going to be like boom, 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 boom. But then you're going to go for a run and it's going to go boom, 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 right? It's going to change the speed of the cardiac cycles. How is this regulated, right? The heart has its own electrical wiring that's referred to as the intrinsic system. Remember those pacemaker cells in the nodes? They're self-excitable and autorhythmic. So a heart, right, when we remove it from a transplant patient, it continues to beat on its own. Well, the nervous system, and endocrine system, they modulate the heartbeat through the extrinsic system. This is just the regular beat, right? But by regulating autonomic activity, the neurons, and the hormones, the body can fine tune the rate of homeostasis as your cardiovascular demands will change. So that will adjust your cardiac output. So all this to say is the nervous system actually has a little bit of a leash on the heart, and it can go and switch up and turn on the volume, up the volume of the heartbeat. Remember I told you the extrinsic system will tell the heart to beat a bit faster or a bit slower. The pace itself is generated here, but how do we modulate the heart rate? That would be the extrinsic system. The nervous system and endocrine system can go and influence the heart's beat, the heart rate. Secondly, you could influence the stroke volume, and that would change your cardiac output, right? If for every beat, let's say you had the same heart rate in situation A and B, but in situation B, you expulsed more blood per beat. That would increase cardiac output. More blood would be sent to the tissues and flow to the tissues. So what is stroke volume? We've already discussed this, right? Stroke volume is the amount of blood that's ejected out of the heart, right? So over here, whatever you collect in your little wine glass that comes out of, let's say, the aorta, right? This amount that got ejected, that's your stroke volume, right? And so the volume of blood ejected during ventricular systole, right? And that equals the end diastolic volume minus the end systolic volume. So the end diastolic volume at diastole, when the ventricle was relaxed and collected all this blood, right? This was your end diastolic volume, EDV, also known as the preload. It's what gets preloaded into the ventricle before it contracts. So it's the amount of blood in the ventricle after diastole, after filling, minus, right, the end systolic volume. This over here, after my ventricle has squeezed out all of this stroke volume, what is left is called the end systolic volume, ESV. That's the afterload. After I have ejected all this blood, what is the load that is left in the ventricle, right? Not 100% of the blood in the ventricle gets ejected. So it's the amount of blood in the ventricle after systole, after ejection. And as you can see, right, here's how much blood was in the ventricle. All of this stuff, right, got expulsed out. That's your stroke volume. So this over here is the stroke volume. The end diastolic volume minus the end systolic volume is your stroke volume, right? So here you see this graph, if it helps you. If it doesn't, don't worry. Here's a graph where you see the volume in the ventricles over time. And you see that the volume over here, right? The ventricle is collecting all of this blood, and it reaches a plateau. That's your end diastolic volume. That's the most volume that's in your ventricle when it's relaxed. And then it will squeeze, right? And exit out all of this blood, actually exit it out into the arteries, right? And once it's done squeezing, whatever's left, right? This amount over here is your end systolic volume. At the end of systole, how much was left? And what I need you to know is that this amount that got ejected is called the stroke volume. It's the end diastolic volume minus the end systolic volume, okay? So that's the idea of the stroke. What is the stroke volume? And I told you, if we eject more blood out, our stroke volume increases. That will increase cardiac output. If we eject less blood out, less blood goes to the tissues, less flow, less cardiac output, right? So stroke volume could be altered actually in 3 different ways. The first way you could affect stroke volume is called what we call the preload. This is the degree of stretching of the ventricular muscle during diastole. How can we, the preload, right, is that end diastolic volume. So over here, here's our cardiac output. We said the heart rate could affect cardiac output, right? But there's 3 ways to affect the cardiac output using stroke volume. All of this is the stroke volume. The first one is called preload. How much blood is put into the ventricles? That could be determined by the degree of stretching of the ventricular muscles during diastole. As it's relaxing, if it's really relaxed, it could open up and collect even more blood, right? So increased preload will increase the end diastolic volume. I like to think of preload and afterload as this little slingshot, right? Preload is like charging up the slingshot. So however much you pull back, right? However much the ventricle relaxes to allow that blood to come in, that will be what can alter it. So it affects the ability of muscle cells to produce tension as muscle stretches past the resting length. The amount of force during systole increases. So what is it that can alter preload? What can affect how much blood is going to be in here, right? Number 1, the venous return. Preload is how much blood is coming back to the heart. So depending on our circulation and how much is coming back, that could affect the amount of preload. And secondly, the filling time. What if I have a fast heart rate? Then maybe I won't have enough time to fill up my ventricles as much as possible. Whereas if you leave a big space, right, a lot of time between the heartbeats, then you might be able to fill up your heart even more, the stroke volume. The end diastolic volume. So an increase in preload will increase the end diastolic volume, right? It will increase the amount that's at the end of diastole. This increases the stroke volume because more blood can be ejected after. There's more blood in the ventricle, more blood can be ejected. And lastly, it will increase the cardiac output. If I increase my stroke volume, more blood exiting the heart per beat, then there's going to be more blood going to the tissues overall per minute, right? The second one is called contractility. This is the forcefulness of contraction of the individual ventricular muscle fibers. Think of a water balloon with a straw in it. I could squeeze the water balloon softly, or I could squeeze it hard, right? An increased contractility will decrease our end systolic volume. If I contract more blood will be exited and there'll be less blood remaining in the heart, meaning less ESV, less end systolic volume, right? If I contract weakly, less blood is ejected and there's more volume of blood in the heart that stays over after systole. So due to the permeability of cardiac muscle to ions, this can change how much contraction is occurring. So what can alter contractility? It could be regulated by the ANS, the autonomic nervous system, or it could be regulated by hormones, right? That can affect the contractility. So what I need you to know is increased contractility means a decrease in the end systolic volume, which means an increase in the systolic volume. And therefore, if I'm ejecting more blood out, stroke volume, if I'm ejecting more blood out per beat, I will eject more blood out to the tissues, right? Per minute. And lastly, the afterload. So the contractility was the ability for my elastic to recoil, right? The afterload is what is the amount that I'm ejecting out of my slingshot, right? So, um, the afterload, it really depends actually on the arterial pressure, okay? So, the tension that must be exceeded by the ventricles to open the semilunar valves. Do you remember I told you this aorta has a very high pressure? We don't want our pressure to be too high because then the ventricles will need to squeeze harder to try to eject blood. Preload is what is bringing in blood into the heart. Afterload is here is the ventricle trying to squeeze out the blood. How much pressure is found in the arteries to burst open the semilunar valves? Remember that the semilunar valves, they burst open over here, right? They need to overcome this. What happens if my pressure, blood pressure, this red line actually ended a bit higher? You would not be ejecting as much blood out because the semilunar valves would only open up a little bit higher, right? So there'd be less time to eject blood out. So afterload, the tension that must be exceeded by the ventricles, here's my ventricles, trying to exceed the pressure, right? To open the semilunar valves and eject blood out the heart. That depends on the pressure in our arteries. So what can alter afterload? Vasoconstriction. If my vessels constrict, or if a patient has hypertension, let's say, like high blood pressure, that could affect this. It'll make the heart work harder to try to eject blood. Or vasodilation, some very relaxing of those arteries, that will make it easier for the heart to pump out. So hypertension increases the afterload, which means less blood goes out, so I have more of an end systolic volume. That means that if I have more systolic volume left over in the heart after contraction, there's going to be less of a stroke volume exiting the heart, meaning less cardiac output, right? And lastly, our topic is going to be ANS regulation, the autonomic nervous system, how it regulates the heart. So, do you remember we talked about the sympathetic and parasympathetic a bit and how they're antagonizing? They fight with each other. Sympathetic is stress and fight or flight, right? The stress response. And parasympathetic is the rest and digest. So the sympathetic nerve fibers and a separate set of parasympathetic nerve fibers innervate the SA node. Oh, here's the SA node and the AV node, right? Here's SA and AV. So there's two separate fibers. Here in this pinkish is the parasympathetic ones. In purple here you see the sympathetic ones. They go to the SA and the AV node. They go to the atrial contractile cells. You see that some of them go to the cells in the atria here. You see that some of them go to the ventricular contractile cells over here, right? And what are they going to do? The two of them, those two nervous system divisions are separate neurons that antagonize each other. The sympathetic nervous system sends signals to increase the heart rate. This purple neuron here, these neurons, they will tell the heart beat faster. This set, the parasympathetic neurons over here, right, they send signals to decrease the heart rate. And they're constantly going to be firing at different levels. And they're going to be fighting signals and kind of confusing the heart, right? And both divisions, they're continuously activated. And they continuously counter each other's effects on the organ until one branch fires a little bit faster and more action potential. So when you're stressed, when you're neutral, right, both of these are firing equally onto the heart. One saying beat faster, the other saying beat slower. And there's this middle ground that's reached. When you're stressed, the sympathetic neurons will fire faster action potentials and get the heart to start beating. This signal will override and predominate and beat kind of the signal of the parasympathetic and make the heart go faster. The opposite is true. When we're resting and digesting, the parasympathetic takes over and overrides the signals from the sympathetic system so that we can beat our heart less fast. So this is called autonomic tone. And this is what determines our heart rate. So autonomic tone is this idea that there's a balance between the sympathetic and parasympathetic activity at rest during homeostasis. It's the idea that physiological values describing the autonomic tone of a dually innervated organ is determined by both divisions. For example, the resting heart rate generally sits at around 70 to 80 beats per minute. What is it that produces this value of 70 to 80 beats per minute? It's a fight between two teams. There is the heart activity that is downregulated by the parasympathetic system. These neurons, parasympathetic neurons, say, hey, quiet down. And these sympathetic neurons go to the heart and say, hey, speed up, right? That's the sympathetic neurons. And so that's what an autonomic tone is. The heart stays at 70 to 80 because these two branches are firing. And this is the tone, the autonomic tone, the baseline activity of the heart based on these two signals that reach some kind of a consensus. So I really like this graph over here because here you see at rest, what's interesting is that at rest when we're calm, right? Actually the parasympathetic level is very high. We're at rest. This is very high. The sympathetic is very low. Heart rate in beats per minute for females on average is about 72 to 80 beats per minute. And for males, it's about 64 to 72 beats per minute. When we sprint, right, we go for a run, our heartbeat increases drastically, 145 beats per minute, because the sympathetic branch peaks and goes much higher compared to the parasympathetic branch, and this fires more relative to the parasympathetic. Then we stop running, and you see that our heartbeat goes down to 120 because this sympathetic tone goes down. This parasympathetic branch starts to increase. And then 30 seconds into recovery, that we've stopped running, our heartbeat goes even lower and our parasympathetic tone starts taking over even more. Our sympathetic one goes down a lot. And then that's like as we start getting back to rest. Interestingly, if we have a lesion to the spinal cord, right, we will see that our heart rate is about 60 beats per minute because the sympathetic fibers actually go through the spinal cord. So that sympathetic tone will be gone, and we have this maximal parasympathetic tone to our heart. So we actually have a much depressed heart rate if we have some kind of spinal cord injury because the sympathetic tone is cut out fully. Where's the control center for autonomic signaling? The cardiovascular center of the medulla oblongata, this is this brainstem region of the brain, is the headquarters for the autonomic nervous system for cardiac control. There are cardio-inhibitory centers in this brainstem of the medulla, right? There's a medulla in the brainstem. And this is part of the parasympathetic nervous system. Cardio-inhibitory will inhibit, right, the cardiovascular system. So here you see this parasympathetic nerve going through the vagus, exiting out to decrease the heart rate, right? It sends signals to decrease heart rate and decrease stroke volume. Cardioacceleratory centers in the medulla is part of the sympathetic nervous system, and it will send out these signals to increase the heart rate and increase the contractility, the stroke volume, right? The vasomotor nerves, right? These are areas of the medulla, the vasomotor regions. They will go out and through the sympathetic nervous system, they send signals to blood vessels to vasoconstrict. This too will have an effect on the heart, right? Because this affects our peripheral blood pressure. So, how does this work? What is the autonomic nervous system's effect on pacemaker cells? The sympathetic and the parasympathetic nervous system, they will synapse, make connections onto pacemaker cells and change their ionic permeabilities. They will change the opening of channels and let more ions flow or less ions. And depolarization or hyperpolarization will affect the heart rate through changes in the rate at which impulses are generated. So let's start over here with in the medulla, right? In the brain stem. Here's the cardioacceleratory center. It sends, let's say it fires. Bzz, bzz. It sends down a signal through the spinal cord out through this sympathetic preganglionic fiber onto this secondary postganglionic neuron, and it will go to these regions over here and tell the heart to speed up, right? So from the thoracic nerves of the sympathetic chain ganglia, the sympathetic nervous system will release, over here, it'll release a bunch of norepinephrine on the pacemaker cells over here, and depolarize them to increase the heart rate, right? It'll tell the heart to beat faster. So here's the neurons, and it will release norepinephrine to tell it to do that. For the medulla and from the medulla and passing through the vagus nerve, here are the cardioinhibitory centers in the medulla, and it'll go down the vagus nerve. This is a cranial nerve. It will reach the pacemaker cells, and it'll release acetylcholine onto them, which will hyperpolarize those cells and decrease the heart rate. That'll make them beat less fast, right? So that's acetylcholine that's released onto these pacemaker cells. So you really see that there's two divisions of the autonomic nervous system that is changing the heart rate. Just some other things: chemical regulation of the heart, hormones that affect the heart, epinephrine and norepinephrine from the adrenal medulla increase the heart rate and increase contractility. Thyroxine directly increases the heart rate and enhances the effects of norepi and epi. Ion concentration, normal intra and extracellular ion concentrations need to be maintained for normal heart functioning. If your plasma electrolyte imbalances occur, right, if you don't have the right amount of ions in your plasma, in your blood, then that could have an effect. Hypocalcemia, if you have low calcium, that depresses the heart. Hypercalcemia, high calcium, increases the heart rate and contractility. Remember that calcium has a whole effect in muscles on troponin and tropomyosin, blah, 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 right? So the same thing for heart muscle, right? The calcium has an important role. Hyperkalemia, too high levels of potassium alters the resting membrane potential and can lead to a heart block and cardiac arrest. Hypokalemia, low potassium, results in feeble heartbeat and arrhythmias. Other factors that regulate our heart rate: age. Fetus has the fastest heart rate, and then it declines with age. Sex. Females usually have a faster average heart rate than males. And exercise. Increased heart rate via the sympathetic nervous system. Trained athletes usually have a lower resting heart rate, actually. And lastly, body temperature. An increased heart rate with increased body temperature, since there's an increased metabolic rate of cardiac muscle cells. That is what's happening. And that's all for this lecture.